auspicious morning to one and all present here. I take immense pleasure in welcoming everyone to the International Colloquium on Healthcare and Medical Engineering, Research, Innovations and Applications, organized by IEEE EMB Society, Madras Section, and IEEE Student Chapter, KH Rangasamy College of Technology. Let's start this marvelous occasion in a divine manner with a prayer song.
गानचतुराय गानप्राणाय गानान्तरात्मने गानोषुकाय गानमट्टाय गानोत्सुकमनसे गुरुपूजिताय गुरुदेवताय गुरुकुलस्थायिने गुरुविक्रमाय गुह्यप्रवराय गुरुवे गुणगुरवे गुरुदैत्यकलाचित्रे गुरुशर्मशराराध्याय गुरुपुत्रपरित्रात्रे गुरुपाकण्डकण्डकाय. Thank you.
The foundation of KS Rangasamy College of Technology was laid in the year 1994. KS Rangasamy College of Technology offers 13 undergraduate and 9 postgraduate courses in addition to MS and PhD programs in different science, engineering, and technology disciplines. 7 UG programs are accredited by NBA under Tier 1 category. Electronics and Communication Engineering Department was started in the year 1994 and has been accredited by NBA under Tier 1 category. The department has active research projects in current technologies like AI, IoT, data science, etc., with applications in healthcare and agriculture. The total fund received from various agencies is 88 lakh rupees. The objective of this colloquium is to bring research scholars from academic institutions to discuss and share their research works in the field of healthcare and medical engineering.
It is necessary to learn from the past, live the present, and welcome the future. Now I call upon Dr. P. Kumar, the head of the Department, Electronics and Communication Engineering, KS Rangasamy College of Technology, to welcome the marvelous gathering.
Thank you, MOC. Hope I am audible to everyone.
Yes, sir.
Thank you, ma'am. Very good morning. Warm greetings from KS Rangasamy College of Technology. Respected Chairman, Line Dr. KS Rangasamy MJF, KSR Education Institutions. Respected Vice Chairman through R. Srinivasan, KSR Education Institutions. Respected CEO, Dr. K. Tyagaraja, KSR Education Institutions. Distinguished Chief Guest of today's inaugural function, Professor Alistair McEwan, Ainsworth Chair of Technology and Innovation, the University of Sydney, Australia. Respected Principal, Dr. R. Gopalakrishnan, KS Rangasamy College of Technology. Respected Dean, School of Electrical Sciences and Chair IEEE EMBS Madras Section, Dr. K.B. Jayanti. Secretary of IEEE EMBS Madras Section, Dr. B. Bhanurega. Branch Counselor of IEEE Student Branch, KS Rangasamy College of Technology, Dr. C. Rajashekharan. Ladies and gentlemen, dear colleagues, I am pleased to welcome you to the International Colloquium on Healthcare and Medical Engineering, Research, Innovations and Applications inaugural function organized by IEEE EMBS Madras Section in association with KS Rangasamy College of Technology, IEEE Student Chapter. It is a great pleasure for me to welcome the participants from all over the world who have joined here to exchange experience and work together on the exciting field of medical engineering and its applications. I first wish to extend sincere thanks to IEEE EMBS Madras Section for collaborating with KS Rangasamy College of Technology and hosting such a wonderful event. Thank you. It is high time that such an event took place to highlight the remarkable contribution which medical applications make in so many areas of our lives. A glance through the list of presentations planned for the next 2 days reveals the amazing diversity of these applications. I would like to render my sincere thanks to the plenary speakers of today's program, Professor Nigel Lowell, Graduate School of Biomedical Engineering, Australia, Ajay Anand, Georgian Institute of Data Science, University of Rochester, USA, Professor Celia Sahanas, Bangladesh University of Engineering and Technology, Bangladesh, Dr. Rafai Choi, Swinburne University of Technology, Melbourne, Australia, Professor Ganesh Noy, Flinders University, South Australia, Professor Michael Fu, Cleveland Functional Electrical Simulation Center, which is a consortium of 5 universities, and Ms. Sazia Nasreen, PhD Research Scholar, IIT Kharagpur, India. They have readily accepted our invitation amongst their busy schedule. Thank you. Before closing, I have to extend further thanks to our IEEE student branch of KS Rangasamy College of Technology for taking immense efforts to organize such a wonderful event. Let me now close by wishing you a delightful and stimulating day. Thank you. Over to Amos.
Thank you, sir. A leader is the one who knows the way, goes the way, and shows the way. Now, I invite such a leader, Dr. K.B. Jayanti, Chair, IEEE EMBS Madras session, to introduce the theme of the colloquium.
A very good morning to one and all present here. A warm welcome to the plenary and keynote speakers who have readily agreed our invitation and accepted to deliver their talk in this colloquium, with special mention to Professor Alistair McEwen, Ainsworth Chair of Technology and Innovation, who is providing the inaugural address, and all the participants who have registered to attend this colloquium from across the globe in various time zones. At the outset, I would like to thank the management, chairman, vice chairman, and the principal, Dr. R. Gopalakrishnan of KS Rangasamy College of Technology, for providing support to host this event. IEEE Engineering and Medicine and Biology Society is the world's largest international society of engineers who work in biomedical domain. Engineers who design the electrical circuits that make the pacemaker run, create the software that reads an MRI, and help develop the wireless technologies that allow the patients and doctors to communicate over long distances. This colloquium is brought in now with the objective that this field of biomedical engineering has promising careers to the undergrad students who really want to contribute to the society. And engineers and researchers in this area are the link between science and life science, creating innovations in healthcare technology for the benefit of all humanity. We have brought in researchers from across the globe who are working in various domains in healthcare and medical engineering, and I believe all the research scholars and the PG students and the undergrad students who are attending this colloquium will definitely get a benefit of how to proceed with their career in case they choose biomedical engineering. With this note, I thank everyone for the opportunity given and for joining us today in this session. Thank you. Thank you, one and all.
Thank you, ma'am. Honor is simply the morality of superior men. Now, I graciously invite such a leader, Dr. R. Gopalakrishnan, Principal, KS Rangasamy College of Technology, to address the gathering.
So, thank you, MOC. Respected Chairman, Line Dr. K. Rangasamy. Respected Vice Chairman, Mr. R. Srinivasan. Beloved CEO, Dr. K. Tyagaraja. Today's the chief guest and the person who will be inaugurating this particular event, Professor Alistair, Head of the Department, Dr. P. Kumar, and the Dean of School of Electrical Sciences, Dr. K.B. Jayanti, and the IEEE Coordinator, Dr. Rajshekaran, and other plenary speakers, invitees, faculty members, and my dear student friends. Very good morning, all of you. I'm really happy to be here to this inaugural function of IEEE Madras section organizing Engineering and Medicine Biology Society, EMBS, as we call the today's international colloquium on healthcare and medical engineering, research, innovations, and applications. So it would be happening for today and tomorrow, 24th and 25th of September 2021. At the outset, I must thank Professor Alistair for accepting our invite and joining here for the inaugural address. Thank you very much, Professor. And it's a really great pleasure and honor for us for having such a wonderful personality for this particular inaugural address. My sincere thanks to you, and the thanks goes to the IEEE Madras section for permitting us to organize this particular event, because as our dean has rightly mentioned, it is a need of the hour. And my thanks goes to other plenary speakers. Today and tomorrow, they will be giving a lot of insights to our budding engineers, researchers, and other people as well. So coming to the 2 days event, why we have to have this event, the engineering, we call it as engineering and medicine and biology society. That's what EMBS is. We are probably very great initiatives been taken by Dr. Rajshekharan and the Department of ECE. I can name many things. Even the COVID time, we visited nearby schools and created a lot of awareness. Not only that, we also tried to come out with some of the applications in our IoT Center of Excellences, Mr. Raghuvaran and other people. So here today, the topic healthcare and medical engineering. In fact, normally we call it as a biomedical engineering. Probably Professor agree. So in that, what are the research, innovations and applications are happening? So earlier, we were cautious about health, our own health. The care of our own health was very less. But whereas last 1.5 years, the healthcare has tremendously changed. It has created a huge impact, because if you talk about GDP, if you talk about the new medicines, now we will be able to mention about so many drug companies. Say for example, if Pfizer, Bharat Biotech, and like that, so many drug companies we come to know. Earlier we will be knowing only few companies, but now we come to know who are there, the players available in worldwide. That's one thing. The second one, the kits, we call it as combo kits. Say for example, if you take Amazon or any the e-commerce vendors or e-commerce companies, they come out with a lot of combo kits. It has oximeter, pulse oximeter, and thermometer, and other relevant to whatever. Again, it's all health related aspects. And earlier we used to wear the Fitbits. It would be like a minimum cost. So people were not that cautious. But last 1.5 years, if you look at the Fitbit and smartwatches, and people wanted to go with a lot of smart applications pertaining to healthcare. So it all shows that how the healthcare industry is growing up, and more than that, how the biomedical engineering, that's what engineering is helping the healthcare. So now we name it as a biomedical engineering. So here it is not only relevant to drug, it is more of relevant to tech. By using the technology, we are coming out with a lot of applications. That's what the research and innovations and the applications. So actually, it has supposed to be idea, innovation, and probably the research will be in between these two. So that's what we are normally inculcating this to our students. Probably I request the researchers and the students, because we have at KS Rangasamy College of Technology, we normally insist our students think and make it as an idea. Then for the idea, we will be helping you with the support of professors like Alistair, and different forums are there. Obviously, that is what our objective. In fact, we have a IIS, Institution Innovation Cell, and even we are very much keen in supporting our students, converting their projects into products. That is what our objective this year, particularly the final year projects. We are insisting the students to go for solving the societal problems, not taking in some paper, solving it. It's all there. What we are going to do, something new. So you please concentrate more on the innovations. Innovations, it's not you have to come out with some new inventions. That's not required. So see, for example, the same healthcare, there are a lot of parameters to be measured. So what can be best? What best you can do? What is existing? So understand that, and we can support the society. Being an engineer, we have to support the society. That is what I used to insist. The science and technology to solve the social problems. So that's one of the area which normally we are keenly highlighting it. And here, any application development, yes, it is possible, because we have Idea Lab. Probably October 1 onwards we are going to inaugurated. So the objective of this Idea Lab, come out with the idea and it can be converted as a prototype. So any idea can be developed as an application at the Idea Lab. This is another area we are working very keenly, so that any student particularly it is not only for the final years. Even the students of first year can come out here. They can walk in, and our mentors will be there so that they can convert as a prototype. So that's the objective. So we have to explore. So obviously engineering, a lot of hands-on is required, particularly this healthcare. See, one last point I want to tell you, probably a professor also knows it. You just take last 8 months, the usage of chips, because it is now worldwide. People are struggling to get chips. So it's all embedded chips I'm talking about. So again, that is where another area it is picking up. So whatever you wanted to know, algorithm, anything you can embed for any particular application, that area is also it's picking up because of lack in the chip manufacturing. Otherwise it's not about lack, and it is about a lot of last 8 months people are started using a lot of gadgets, so many computers, laptops, and automation everywhere. It is picking up, so that the requirement for that embedded chips, it has increased dramatically. So that's another area where we can think about it. So overall, I will tell you. So this research, innovation, and application, let us continue our work for the betterment of the society. And no doubt, obviously, if we come out with a really good product, even we will be able to get Nobel Prize. That's what finally I wanted to touch upon. Probably our professor will also agree. And let us come out with some really good innovations, and it can help. It should help the livelihood of the people more than life. Very well how precious it is. So obviously the technologists, it is not only the doctors. We are more than doctors. That's what I can finally say. And I once again thank my sincere thanks to Professor Alistair for accepting our invite and joining us. And the next 2 days, let us listen to the speakers and understand. If you have any doubts, that's very important. It should not be like only the inputs given by the professors. And wherever we have doubt, anything coming in out of our mind, let us interact, so that we'll open, we'll get an opportunity to understand our doubts. We get clarification from the experts. So finally, I appreciate the department and IEEE student chapter of KSRCT for conducting this type of events regularly. So obviously this will create an impact, no doubt about it. So stay safe and take care, get vaccinated. That's another thing which we have to say now. So get vaccinated and let everyone safe in this world. Thank you. Over to MOC.
Thank you for your enlightening words, sir. Optimism is the faith that leads to achievement. Now I graciously invite Ms. Nalini, second year, Department of Electronics and Communication Engineering, to introduce our chief guest address, Dr. Lester McEwen, Ainsworth Chair of Technology and Innovation, the University of Sydney.
A delightful morning to one and all present here. Success is not final. Failure is not fatal. It is the courage to continue that counts. It's a great honor for me to introduce and welcome our eminent chief guest in this auspicious occasion. On behalf of all gathered here, I cordially welcome to our honorable chief guest, Dr. Alistair McEwen, Ainsworth Chair of Technology and Innovation, the University of Sydney. He is a senior member of the Institute and Electrical and Electronics Engineers, Engineering in Medicine and Biology Society, and Technical Committee of IEEE Biomedical Circuits and System. He is an aspiring motivator for the youngster today. In 2011, he was awarded a Microsoft Future Fellowship, the first Australian to be recognized with this award for his work in developing electronic devices to diagnose heart attack and stroke early. In 2017, Associate Professor Alistair McEwen was appointed as the Ainsworth Chair of Technology and Innovation in the Faculty of Engineering and Information Technologies. Professor McEwen is passionate about engineering technology innovation to solve problems of real clinical need. His specific research interests include development of fetal and placental monitoring technologies using pregnancy, including ultrasound and electrograms for integrations with neonatal and infant monitoring and brain-computer technology with audiovisual feedback. Professor McEwen has a world-class record of research and collaborative work in biomedical engineering, electronic and signal processing. He has over 120 referred publications, 8 patents, more than thousands of handle citations, and has obtained over $7.5 million in competitive research funding. Our speaker is a great motivator who has the vision to create an impact in the student career. We are privileged to have such an eminent and prominent persons here among us. Sir, we are really thankful to you for making your presence today despite your busy schedules. We wish all your attempts would result in a great success. Thank you, sir.
Thank you, Nalini. If your actions inspire others to dream more, learn more, do more, and become more, you are a leader. Now I cheerfully invite such a person who has accomplished success and our most valued guest of today's session, Dr. Alistair McEwen, Ainsworth Chair of Technology and Innovation, the University of Sydney, to take over the session.
Thank you so much. Thank you for the invite, and I'd love to thank all the organisers of this wonderful meeting and all of the speakers who've spoken before me, and I'd like to echo again their thanks to everybody involved, and it's a great pleasure to meet everyone who's joining from around the world. I'd like to start off thinking about something a little bit different. We can get inspiration for new innovations from looking at the world around us. So here is a beautiful picture of Sydney Harbour. I hope you have the chance to visit one day. And you can see two animals here that live in Sydney. And you may have seen a photo of these animals before or heard about them. Many people I talk to when I travel around the world telling me that they know about the dangerous animals in Australia. And I'd like to tell you that here's another couple of dangerous animals. And just to get you thinking, perhaps you would could think about which of these animals is more dangerous. One is the platypus, which you may be familiar with, that has a face like a duck, but a body like a beaver. And the other is a stingray. And both of these animals use electrical sensing to find their food. And in the case of the electric ray, they use a higher voltage to capture their food. The animal that's more dangerous is actually the friendly-looking platypus here. He has a toxin, if it's a male, that can put you in hospital for quite a while, but it's also being used for medical treatments. He has electrical sensing on the front of his bill here, but also mechano-vibrating sensors, so sensors similar to what we see in ultrasound. So this amazing animal, we can take inspiration from that animal and think about how it could, say, combine electrical sensing like ECG and ultrasound and be able to pick up perhaps things like tumours or other things that we're interested in the human body in a new way. So my research really is trying to take some of this inspiration, and one way we look at it is to think about the whole electromagnetic spectrum that we have at our disposal. And many of the medical devices that are developed and we use different frequencies in this range. But we haven't really explored all of them. We use ultrasound, we use higher frequencies for X-ray, but there are many more that we can use. There is a picture here of a device we use to measure the amount of fat in babies with light. There's shown here a pacemaker where we're picking up the small voltages from the heart, but we can take that kind of energy that we use to pick up a heartbeat and increase that, increase the voltage again to make a pacemaker to pace the heart. We can also increase the voltage even further and open up the cell membranes to introduce drugs, or we could heat the tissue even further to cause a very specific electrosurgery. And that's what we're doing here with a catheter, a cardiac catheter that uses microwaves to heat the tissue at a distance so that we don't cook the blood and possibly create an embolism that could lead to a stroke. So we heat the tissue, the heart tissue, away from the catheter, and in that way the device can be more safe. I was asked to talk a little bit about future careers for all of the wonderful undergraduate students who are joining. And just as the previous speakers mentioned, we have a whole new world now with this pandemic. Of course, there are many medical devices, even vaccine manufacture, that's becoming very, very important. And I think we're going to see this only increase. It's clear that governments have decided that it's worth pausing economic development to deal with the pandemic because it is the bigger problem, and that sentiment maybe will be continuing, and we'll be putting more value in the healthcare that we can deliver, and particularly with technology, because obviously the technology, it's not often said in biomedical engineering, but with technology we can obviously address much larger problems than single doctors can on their own. You know, with technology and doctors we can solve many problems. I think we'll see this, as was mentioned, with the technology companies looking at kits that involve wearables. Many people looking at computer vision now with telehealth. A lot of healthcare delivered just like we're delivering this this session in a Teams teleconference. Assistive technology, and here's a really big opportunity for you. Remarkable is a startup accelerator in assistive technology run here in Sydney, but anybody in the world can join this program over Zoom and over Teams calls. We'll see medical device regulation is becoming more harmonized around the world. So devices, developed in India can be used right around the world. And you can get inspiration from some of these challenges that I've listed here, from Johnson & Johnson, from a company called 9 Sigma, which deals with lots of companies' open innovation problems. They're looking for solutions. And the Gates Foundation, which you might be familiar with. I am a chief investigator in this ARC Training Centre for Innovative Bioengineering. And here we're looking at different ways of interfacing with the human body. So using electrodes that are 3D printed and then placed on the skin like a tattoo that better conforms to the skin, like the one shown here on the hand. And electrodes that can be introduced to the brain in different levels. So deep within the brain, like a deep brain stimulator that's used in Parkinson's and now depression. Electrodes that are penetrating just slightly into the brain underneath the skull, which is similar to what Elon Musk's Neuralink company is doing. Electrodes placed on the surface of the brain, and electrodes placed on the skull under the scalp, and of course electrodes on top of the scalp as well. We're developing new electronic circuits that can deal with this type of implantation and pick up the different types of signals that you see at different levels of invasiveness. And there's many applications described around here for computer gaming, like what Michael Fu will talk about. Assistive technologies for the foot, for the hands, even to help someone who has spinal cord injury walk again, and also prosthetic devices like the hand shown here. Just some news from today. This news came out today that a company run by a startup company run by 2 PhD students in my lab just received a large amount of second funding for their product, which is about monitoring resuscitation given to very small babies very early on in life. This can be used right around the world, and it gives the user a display, and it shows them, like this green light here, that everything's going okay with the resuscitation that they're providing. But if it's not, the light will change color, either orange or red, and that will tell them that there's a risk of the baby receiving too much pressure, which could lead to a brain injury and cerebral palsy, which I'll talk about in my other talk. or if they receive too little oxygen, then their brain might not get the oxygen it needs, and they may also lead to a disability or death. So this is a really scalable device that's solving a worldwide problem. So I'd like to thank everyone again. It's wonderful to meet you all and enjoying this session. And please get in touch if you'd like to hear more. Thank you.
Thank you, Professor. Thank you very much.
Thank you, sir, for the marvelous and knowledgeable. Sorry, ma'am.
Yeah, please continue.
Yes, ma'am. Thank you, sir, for the marvelous and the knowledgeable session. And no matter what language we speak, a kind and smiling thank you will always help to speak to everyone's heart. Now, I call upon Mr. K. Raghuvaran, Assistant Professor, Department of Electronics and Communication Engineering, KS Rangasamy College of Technology, to propose the vote of thanks.
Thank you, MOC. Happy morning to all. On this auspicious day, on behalf of IEEE Engineering in Medicine and Biology Society Madras Section and KS Rangasamy College of Technology, Tiruchengodu, India, I would like to thank our Honorable Chairman Dr. KS Rangasamy and our Vice Chairman Mr. R. Srinivasan for providing all facilities and motivation to us for organizing this international colloquium. I extend my heartfelt thanks to our Chief Guest, Professor Alston McQueen, Ainsworth Chair of Technology and Innovation, University of Sydney, for inaugurating this auspicious event. I extend my heartfelt thanks to all our speakers for accepting our invitations and ready to spend their time and share their knowledge. I would like to thank our beloved Principal, Dr. R. Gopalakrishnan, and Head of the Department ECE, Dr. P. Kumar, for their motivation and timely support for organizing this event. An event like this cannot happen overnight. I express my gratitude to Dr. K.B. Jayanti, Chair, IEEE EMBS Madras Section, for continuous effort and dedication for this international colloquium. I extend my special thanks to co-chairs of this event, Dr. B. Bhanureka, Secretary, IEEE EMBS Madras Section, and Dr. C. Rajashekharan, Branch Counselor, IEEE Student Branch of KS Rangasamy College of Technology, for their continuous support for organizing this event. I would like to acknowledge our gratitude to all the members of IEEE EMBS Madras Section, faculties and students of KS Rangasamy College of Technology, who are all acted as a backbone for smooth organizing of this international colloquium. Thank you all. Over to MOC.
Thank you, sir.
The inaugural ceremony comes to an end, and we will start with the sessions of plenary and keynote addresses. And the schedule of the various speakers for plenary talks and keynote sessions are displayed herewith, and we will be starting at 10:35 with the first speaker. Thank you.
Ma'am?
Yes, Raghu?
Ma'am, Mr. Nigel sir has joined, ma'am.
Okay. Professor Nigel Lowell.
Good morning, everyone.
Welcome. Welcome to the International Colloquium. Thanks a lot for joining us in time. And just now we completed the inaugural address, and we have Professor Alistair McEwan here on this link with us. Shall we start the session, Professor?
Absolutely.
Yeah, I'm happy to welcome Professor Nigel Lowell on behalf of EMBS Madras Section and KS Rangasamy College of Technology to this International Colloquium on Healthcare and Medical Engineering, Innovations, Research and Applications. Professor Nigel Lovell received his BE Honours and PhD degrees from the University of New South Wales, Sydney, Australia. He is currently head of the Graduate School of Biomedical Engineering, University of New South Wales, where he holds a position of Scientia Professor and is also Director of the recently established Tyree Institute of Health Engineering. He has authored 300 plus journal papers and been awarded over $100 million in R&D and infrastructure funding. His research work has covered areas of expertise ranging from cardiac modeling, telehealth technologies, biological signal processing, and visual prosthesis design. Through a spin-out company from University of New South Wales, Telemedcare Limited, he has commercialized a range of telehealth technologies for managing chronic disease and falls in the older population. He is also one of the key researchers leading an R&D program to develop in Australia a retinal neuroprosthesis or bionic eye. He is a fellow of 7 societies, learned academies throughout the world. For 2017 and 2018, he was the president of the IEEE Engineering in Medicine and Biology Society. And we are very happy to have you here with us, Professor. And his talk today is scheduled, the title of his talk is Innovations in Wearable and Plantable Bionics. It's all about the interface. The session is open to you, Professor, for the next 30 minutes. Welcome you. Thank you.
Thank you kindly. It's an absolute honour to be here. And I just got, sorry I couldn't be here for the beginning of the opening, but I just heard Alistair's final couple of slides, and it actually is a great segue to my talk, because he was talking about his Australian Research Council Training Centre, and he was talking about interfacing between sort of the biology and the technology, and I think that's an absolute key in the advancement of the field. And the field that we're in is actually it's truly an exciting field at the moment. Nearly every day we hear of amazing advancements, and it's becoming ever more interdisciplinary as well, and it's impacting our life in all different ways, including relieving the burden of disease and improving the quality of life. So there's a picture of my building at the top of the screen. I can't go there, as Alistair well knows. We're in lockdown in Sydney, but luckily I live near the beach, so I can go there. And once we come out of lockdown, I'd actually welcome you to visit Sydney and visit our labs as well as the sites of Sydney. And hopefully that will happen next year. But today I'm going to relate to you some tales of medical device translation. I'm going to focus on one area of research work in my laboratory around a vision prosthesis or a bionic eye. I'll talk about the work and highlight why the field in general has reached a sort of an impasse. And I'll try to identify some of the challenges that require the introduction of novel and disruptive technologies to permit advancement and sort of to permit passage beyond that impasse. And I think quite simply for implantable neuroprosthetics, which is what I'm going to focus on today, the single most important advancement needs to be in re-engineering the neural interface. It's all about this interface. So I'll spend quite a bit of time in my talk highlighting how we can improve the interfacial characteristics of implantable bionics. So in general, the work in our laboratory is in 2 fields, biomonitoring, and I will talk a little bit about biomonitoring at the end, because interfacing is also essential in biomonitoring. But as I said, I'm going to focus this talk mainly on the second area of work in our lab, and that's around neurobionics. So we develop control systems for heart pumps with colleagues at the University of Sydney. We're working on a blink restoration implantable device. We do sensory restoration, so that's going to be the main topic of my talk today. So a bionic eye or a neural retinal prosthesis. And I'll also touch briefly on an augmented cochlear implant. And then I'll talk a little bit about next generation neural interfaces, these disruptive technologies we need to really advance the field. And I'll talk about just 2 of the projects in our labs around gene electrotherapy and optrodes, or optical electrodes. So let me begin and just give you a little bit of a sort of a background to the whole concept of biology technology interfaces. So I think one of the biggest BME challenges is to create a paradigm shift in bionic eye device design, and to do that, I think we need to focus on this concept of next generation bionic interfaces. So our challenge in this case is to re-engineer the nervous system and the brain pathways to create seamless connections between the biological systems and I guess their synthetic replacements, restoring the interaction and perceptions of the world. So we do that through the connections, interactions and perceptions, which are all critical aspects of how we as humans develop, function and exist in the world. Now, sadly, in the case of disease or injury, people lose certain functional abilities, but luckily with technology, there exists medical device therapies to restore limited aspects of this lost or impaired function. It's a huge market. So the neurotechnology market is about $20 billion with a 13% growth and over 500,000 patients implanted every day. And I guess our end goal here, the ultimate goal, is to create a bionic device technology that completely restores a patient's ability in a manner which is indistinguishable or even augments normal human function. So that's where we're at. And for those of you who are old enough, if I can borrow from Steve Austin and the $6 Million Man, a crisis truly has and still does exist in this bionic interface world. So let me give an example. Here's a paper from a couple of years ago, Nature paper, paralyzed subject with one of these implanted UTAR arrays of 100 metal electrodes for recording in the motor cortex of the brain, and non-invasive stimulating electrodes you can see there attached to the person's arm. Basically this is to sort of use the brain signals to try to reproduce motor function. Here's another example. This is a current ECOG array that they use for mapping epileptic seizures, and this is current clinical practice. You can see the wiring issues that we have in both those scenarios. I guess in terms of this neural interfacing challenge, I guess the question is how do we do the interfacing now? And it's pretty much like that. We basically probe, push, or poke a metal electrode into excitable tissue. It's not very elegant. So I guess on one side of the spectrum we have this sort of channel limited or static solid, metallic, and sort of macroscopic world of electrodes. And then on the other side, we've got this sort of gray, squishy, multi-channeled, sort of dynamic, ionic world of neural biology. And somewhere in the middle, we have to meet when we're trying to do the interfacing. And I guess our premise is that we have to build that interface from both sides. So we need to bridge that gap by coming up with better technologies, but also better approaches in terms of molecular biology, and looking at the sort of the cytotoxic response, making sure the materials are biocompatible, etc. So why do we need to bridge the gap in the first place? I guess let's take an example of here the cochlea. So here we have a cochlea, a bionic ear, and it produces very impressive results. But if you look at it, there's a big mismatch there. On one side, the technology side, we have 22 platinum metal electrodes, but nature, for very good reason, gives us 30,000 spiral ganglion cells to sense sound. So there's this large mismatch in the number of channels, and because of that mismatch, we can't effectively decode sort of pitch and people with cochlear implants really have difficulties enjoying music, for example. And this sort of mismatch applies to all the bionic devices of today. And I guess the area that I'm most interested in for many years now is in the area of the bionic eye. So I guess if you were to plot functional channels versus quality of life, what you will see down here is that pretty obviously no functional channels, no vision, very poor quality of life. The current sort of state of the art for bionic eye devices is in some respects down here. We've got a handful of channels, of functional channels, and the best we can do is probably motion detection, movement, and mobility work. What we've been working on with colleagues is a sort of 100-channel type device to provide unaided navigation. But truly what we want to get to is these sort of another order of magnitude increase around 1,000 channels, because at that level of resolution we can start to do things like have facial recognition, allow for reading and much improved mobility. And that's where we want to obviously get to in the longer term. So I'm going to talk a little bit about our technology around here, which is the sort of work that I've been doing for nearly 20 years now. Originally starting out with, I guess, a colleague from now at the University of Sydney, who leads biomedical engineering at the University of Sydney, Greg Swanning. So Greg and I have given a number of these sort of bionic eye talks around the world, and despite vision loss having such a huge impact on the lives of individuals and the cost to society, I think we tend to take sight for granted. So here's an example of me I was actually in Vietnam at the time. I was giving a talk in Vietnam. This is a pathway to help to help navigation for those with visual impairments. That's looking one way, and looking the other way down the path, we have things like this. Here's a pathway in Japan, not making it too simple. Here's actually in Malaysia, in Kuala Lumpur, looking one way and then looking the other way. And I don't think you want to be vision impaired in this street in China. So I think that we shouldn't take vision for granted. And part of the reason is that it is a huge problem. It's essentially affects 30 million people throughout the world. 1 in 3,500 people are affected by the most common, or one of the most common blinding diseases, retinitis pigmentosa. And there's no effective effective technology or pharmaceutical approach for restoring vision in those people. So vision prosthesis work has been around for many decades, nearly 50 more years. Here's the first cortical prosthesis from Giles Brinley, where they buried a number of coils under the skin, and then they had an external energizing coil which would sort of inductively induce a current in the coil implanted in the skin, and those coils were connected to electrodes that were then, or a wire to an electrode that was placed over the back of the cortex, the visual cortex, and the person would actually start to see a sort of a spot of light, what we call a phosphene, at about the size of a grain of rice at arm's length. So that was one of the first visual prostheses. But if you were one of these initial 2 test subjects, here's an X-ray and here's an example of the electronics, once again you'd be sort of asking, can't we do the interfacing better? And I guess that's what we've been sort of working on. The technique's still the same. We're still passing a current into neural tissue to try to evoke a response, but we're trying to improve greatly the interface. And we can pass that current using electrodes at various locations. We can put it on the cortex like I just showed you then. We can wrap it around the optic nerve. We can bury it into the lateral geniculate with a penetrating electrode. But most groups, including our groups, place it on the surface of the retina, and we can place it either sort of behind the retina, in front of the retina, or even behind what's called the choroid, which is further from the retina, but it's a much simpler surgical approach, and that's what we've been working on. So here's a picture of how it works in sort of pictorial form. We have an external source of an image source from a camera that's decoded, and that's the signals are sent wirelessly to an implant. It's a two-piece implant, one that's behind the ear that's implanted that picks up the power and the data. There's two wires that connect to the intraocular implant over here. That sort of multiplexes the signal and delivers it to one of 100 electrodes that are placed here on the surface of the retina where the electrode array has been inserted. Those electrical impulses pass actually through the choroid, through the diseased photoreceptors, which would normally convert light into electrical impulses, but in these blinding diseases they've disappeared. And they stimulate these cells here, the retinal ganglion cells, that then synapse or their axonal processes form the optic nerve, and the signals pass to the visual cortex for processing. So that's how we sort of bypass these atrophied or dying photoreceptors. Just checking the time here. I've got another. I'll probably have to speed it up a bit. Sorry. So what we've got here is some inpatient testing with our colleagues down in Melbourne. This is for this suprachoroidal array where the patient was sort of implanted with 20 so electrodes and a percutaneous lead, and we use that to do what's called psychophysics. And instead of just seeing these very punctate and I guess discrete spots of light, we saw these phosphene shapes that were sort of splayed out, and they weren't consistent in intensity. There was sparkling and gradations, and we realized that it's a heck of a lot more difficult than what we thought. So we ended up coming up with ways of trying to improve the way that we injected current into the retina, but also did it in a way that we could do it simultaneously, because if you have to present one spot of light at a time and then sequence it across your electrode array, you just can't form an image fast enough. So that's why we created this design, which is sort of this is more closer to the real piece of electronics that we've made. But on the process, we also were very aware of regulatory, and this is one of the big things that I think that we as researchers have to grapple with, and that is, do we concern ourselves with the translation early on, because that might well dictate what materials we use and how we design the product. And biomedical engineers have truly contributed greatly to the longevity and the safety of implantable medical devices. So this guy is Arne Larsen. Got this slide from actually Greg Swanning, as I said, but Arne was the first person back in the 1960s or late 1950s to receive an implantable pacemaker. A day later, he was the second person because the device failed, and over his lifetime, he received 26 devices, and he had a long life of about 80 years, but that was because initially we didn't know how to make these devices to last a patient lifetime. One of the things we do now is we've set up in Sydney, both at UNSW and at Uni of Sydney, these facilities that allow us to sort of have a mini cochlear-like fabrication facility that allows us to make these devices to safely create capsules to hermetically seal electronics and place them inside the body. So we've got one of the I guess the most sophisticated technologies for using ceramic, layers of ceramic and platinum to encapsulate electronics so it can last in the body for patient lifetime for 50 years. So for our bionic eye, we have to create a 100-channel implant, but we can create a 1,000-channel implant if needed. So not only do we have to worry about hermeticity, but when we're doing these things, there's a whole lot of other tests that I won't go through, but literally including biocompatibility. These are the sort of things you have to worry about if you're trying to not just create something that works on the bench, but you're going to put inside a person to last a lifetime. And as I said, one of the things that we focus on in the design of our devices is novel technologies to allow us to steer current, and we based it on, of all things, the unit Ermatidia of the fruit fly, these hexagonal sort of eyes, and we created our electrodes arrays that way because we found that if we injected current without sort of capturing the charge around that injection site, the current would spread out, and it would just appear as if it, so here we're stimulating 4 sites, but without this sort of return path for the current, the phosphine would just look like one great big blur. But if we actually captured the current using these sort of hexagonal guards, we could create these much more focused and punctate phosphines. And the nice thing about this sort of design, if we came up with clever electronics, is we could paint the scene by just moving it to 7 different locations on that hex surround. So that allowed us to create a very scalable and efficient way of injecting current. So we made electronics to support that, and we came up with some other clever ideas of sharing the return, not just between this local hex, but also a distant monopolar return. And that allowed us to sort of shape the response and trade off stimulus, I guess, lower the threshold of the activation, but also make the penetration of the current, adjust that in terms of how far it's spread out. So we've done that work both computationally, but also in animals, and I don't have time to talk about that, but it effectively means that we can sort of change steer the current in very precise ways onto the retina. And this is work that sort of has been ongoing for quite a while, which is sort of we're sort of at the stage of doing preclinical work, chronic testing in animals. And this is the electronics for the device, and this is the overlaid final implantable device with the 2 parts I spoke about. The part that's implanted behind the ear to collect power and data, and the actual neurostimulator itself. I'm aware of the time, so I should skip it along a little bit here, I think. The next generation of devices, though, is something that, to get to this, to go from 100 to 1,000 channels, though, requires us to really come up with some new and disruptive technologies. It's just not good enough to create more electrodes because, like I showed you before, the current spreads out and smears. We've got to come up with ways of creating a more intimate interface between the biology and the technology. And I'll give you 2 examples of that. One is by using a gene electrotherapy approach. So we place an mRNA or a DNA plasmid in the cochlea here, and then we give an electric pulse, and that causes it to that DNA to be incorporated inside the cells of the cochlea. And the DNA in this case encodes a neurotrophic factor, and it causes neurites to regrow towards the cochlear electrode. So here's a guinea pig that's been deafened in the control side, and here's one that's been transfected, and we can see that we've recreated this neurite regrowth towards the cochlea, towards the stimulating electrode in the cochlea. So this work has been, we've done quite a lot of modeling for the best current pulses to cause that transfection. So just to reiterate, the transfection, to get these DNAs inside the cells, or the mRNAs, we can use a virus, which is like what the AstraZeneca vaccine does, or we can use a lipid, a fat, sort of fatty micelle around the construct, which is like the Pfizer vaccine, but that's very new. but it's not targeted. It goes throughout the whole body. We use an electric pulse to drive the mRNA inside the cell. We've optimized that, and we've shown in years of control versus a treated animal, we've got literally another 12 dBs versus dynamic range, which is huge. And here's the chronic, that's a chronic guinea pig preparation there. And we've started to use that to transfect muscle as well. And I won't go through the video here because of lack of time, but we've got a system that essentially allows us to deliver that into muscle for DNA vaccines and immunotherapy. So what I want to do on the last couple of slides is talk about another form of disruptive technology. So just to finish that previous slide, the gene electrotherapy allows us to basically create more intimate connections between the biology and the technology by causing neurite regrowth. So we're not coming up with a better electrode material now, we're actually manipulating the molecules, performing the molecular biology to cause sort of a more intimate connection. In this case, what we're doing in this second example is coming up with a better technology, an optical transducer to passively record electrical potentials. In this case, I've only got time to talk about the recording action, but we also have a means of doing stimulation along these lines. So the way this device works is essentially that we use a polarized light beam and we shine it through a layer of special liquid crystals that can change its configuration depending on the voltage across these two plates, which act like an electrode or act like a capacitor. And that causes a change in the amount of reflected light, which we can measure. So it's purely a passive process that allows us to sense what's happening here. And here's a sort of another construct of it. So we've designed these systems. We've used it for recording from cardiac electrograms in the synovial node of rabbit tissue. Here's some sample electrograms where the red trace is actually the electrical signal, and the black trace is the optical, the signal from this optrode. The nice thing about the optrode is that we can scale it up, so we can create many hundreds or even thousands of them, and as long as we can come up with ways of feeding in the optical signal, and that's something that the telecommunications industry has a lot of expertise in, then we can just use a single optical fiber with some Bragg gratings to read out from under the tissue, which is, that's work in progress right at the moment. So this sort of work allows us to get up to here, to the next generation technology, we believe. But it is a disruptive technology and it has higher regulatory barriers because we're talking about new materials and coatings, and new, very, very sort of, as I said, disruptive and new approaches. So exciting times, but lots of work to do before it gets into people. So just to summarize, the current technologies allow for hundreds of electrodes, but if we want to go to thousands, we've got to come up with these disruptive technologies. And I wanted to finish off in the last just couple of minutes, talk a little bit about some of our biomonitoring work. So we do a lot of work on telehealth technologies, and once again we have to interface the technologies with the person to assess and manage their chronic disease. So we do wearable devices, smartphone apps, and predictive analytics. And we've been doing this for many years. Here's our initial prototype of a device with my colleague Branko Sella. This was taken through FDA and CE marking, and we've run trials on that throughout Australia. And with these sort of trials, we can show about a $19,000 per year saving, or about a 6 times return on investment using the technology. And here's sort of a control group, and here's an intervention group, and here's where we start to intervene, and we see that the cost of pharmaceuticals and a similar graph for their hospitalizations, that's what sort of predicts those savings. We've shifted recently to make it sort of go with the times and not have a sort of a standalone device, but use the smartphone as the integrator to collect data. And we're running a trial of 2,500 cardiac patients throughout New South Wales at the moment for managing cardiovascular disease. And we've shown just in the initial data that we have about a 40% reduction in hospitalizations after 6 months using these sort of monitoring technologies. And now we're applying that not just to heart, but to COVID and many other areas as well. So, the whole premise of this is to collect all these data from wearables and feed it into our predictive analytics systems to try to risk stratify patients and better manage them to keep them out of hospital. And trying to bring those two sort of topics together, I guess what we're sort of saying is that now implantables even more are starting to have reverse telemetry in them that allows us to send data back to these sort of telehealth systems, and we're working on various variants of those monitoring systems. So the end goal, and this is my last slide, I promise, is that what we're trying to do is integrate the telehealth systems with the by having wireless interfaces, capture reverse telemetry from implantable devices, fuse it with wearable technologies that we've developed, use the smartphone as sort of a patient-centric data integrator, give the tablet to the healthcare worker. And with this sort of technology, we believe that, and we've done some initial health economics on it, we can have a 12 times return on investment compared with usual care. And that's, I guess, our grand vision of interfacing. So on that note, sorry I went a couple of minutes over time. I'd like to thank a whole lot of colleagues, and I don't have time to individually name them, but lots of collaborators. This isn't all my work, it's all a collaborative. Thanks for that, guys.
Yeah, thank you, Professor. That was a wonderful talk on the wearable and implantable bionics. And the session is open for questions. In the meantime, I have a question what would be the battery lifetime of these implantable devices? How frequently should we go for it again?
Okay, so the devices we're making, like the Bionic Eye, don't have any implantable batteries because the revision surgery would be too difficult. So that's a similar thing to a cochlear device. So the power is provided by an inductive link. So it's as long as you've got the external sort of electronics, it powers the device. The other devices, the other implantables, like for instance the pacemakers, implantable defibrillators, and the like, have batteries and they last for many years. But the problem is that as you try to get more data out of them using reverse telemetry to try to help manage the patient's health, it uses a lot more power. So if we were to sort of be monitoring the heart condition using these devices and feeding it back to our telehealth systems, the pacemaker might only last a couple of years instead of 5 to 10 years. So there's a trade-off at the moment around that. And one of the other barriers for advancing the field is to come up with better battery sources.
Yeah, that's fine. So as far as the Bionic Eye is concerned, we do not have a battery. That's great.
We don't need a battery for that.
Yeah, that's a great news. Any questions from the participants?
Participants can unmute your mic and raise your queries.
Yeah, it's always a bit strange talking into the void. I don't know who's out there.
True. And I have another question in the meantime. Like you were telling that it's tested in the sheep. Does it actually perfectly work in humans? Like, what is the idea? Now, where should we test it exactly so that we are confident that it is going to work in human? Any reference for that?
Oh, it's a consuming question, and it's like literally, tens of millions of dollars of expense, and many, a group of probably 20 of us have been working on it for many years. It's really difficult for these sorts of. I think bionic eyes is probably one of the most difficult technologies you could imagine. The visual sense is so complex. Don't know why I ever started out in that field. I should have stuck with something a lot simpler, So look, yeah, and you put the device into a sheep, but the sheep can't tell you much. It can just sort of bleat at you. So you have to do electrical recordings of the brain activity, or you have to do some psychophysics testing. So that's hard. Most of the sheep work we've been doing is safety testing, efficacy at the moment, and we're hoping to go from that to initial human trials with limited functional testing, because we've done a lot of functional testing in other animal species, not chronic testing. So yeah, it's a very long and protracted process. For the augmented cochlear implant that I mentioned with the gene electrotherapy, we've got that into humans now. We've got 6 humans who've had the technology, and it's worked amazingly well. But even that was, each dose of the DNA cost $25,000 just because of the fact we had to take it through this GMP process from Europe. So that's not to say that the final dose would cost much at all, but it's just these initial phases. It just talks to the point that these days, to get things done like this, you have to collaborate with around the world with groups with multiple expertise.
Okay. And this bionic eye actually, is it only for people who have lost the vision completely in both the eyes, or it could be done in one of the eye also?
It's like the cochlear in that they do it they started out in one eye, and it's only certain forms of vision loss. People who have lost their optic nerve, of course, due to glaucoma, it wouldn't work. Because glaucoma, the pressure in the eye chamber causes the ganglion cells to die and the axons, so there's no pathway to the brain. It's only for blinding diseases of the photoreceptors, and that's about half the cases of blindness. So yeah, you can certainly put it in one eye,
Okay. Thank you for your answers, Professor. Any questions? Any questions from the participants? Okay, I think we are done with the session. Thank you, Professor Lowell, for your time and a wonderful presentation. I render my heartfelt thanks on behalf of EMBS Madras section and KS Rangasamy College of Technology for sharing your time and presentation with us. Thank you very much.
Thanks, everyone. Really appreciate it. And it's great. It's a great initiative of you, of the Madras section to put this together. And as an ex-EMB person, I'm really happy to see this activity even in COVID times. So congratulations on pulling together a great event. Thank you.
Ma'am, Banureka ma'am.
Ma'am, good morning.
Yeah, good morning. Welcome, ma'am. I now invite Dr. Banureka to chair the second session of this program today by Professor Alistair McEwen.
Thank you, madam. Welcome, Professor Alistair McCain, and thank you, Professor Lovell, for your wonderful talk. It's really a pleasure to be a part of this international colloquium. I'm honored to introduce the speaker for this session, Professor Alistair McEwen. Madam, am I audible?
Yes, ma'am. You can continue.
Thank you. Thank you. Professor Alistair McEwen is the Ainsworth Chair of Technology and Innovation at Cerebral Palsy Alliance and University of Sydney, Faculty of Engineering and Information Technologies. He is a team leader for biomedical devices and instrumentation and helps lead the Australian Research Council Training Centre for Innovative Bioengineering. Professor McEwen is passionate about engineering technology innovation to solve problems of real clinical need. His research focuses on harnessing advancing technology and innovation to accelerate the search for new and improved treatments and interventions for childhood disabilities and illnesses. Professor McEwen is now identifying priority areas and engaging engineers to help address technology challenges. With teams in engineering and health, he is investigating improved methods of mobility and communication for people living with cerebral palsy using the latest available technologies. For example, he's looking at how we can assist movement using bionics, robotics, and artificial intelligence to help people with cerebral palsy stay better connected to their communities. Professor McEwen is an acknowledged leader. He was awarded a Microsoft Future Fellowship, the first Australian to be recognized with this award, for his work developing electronic devices to diagnose heart attack and stroke early. Prior to this, he held prestigious fellowship positions at University College London, Philips Research, and the University of Oxford. Moreover, Professor McEwen has co-founded two Australian startup biomedical device companies, acted as consultant for biomedical device companies, and has been a member of scientific advisory boards. Professor McEwen has a world-class record of research and collaborative work in biomedical engineering, electronics, and signal processing. He has over 120 refereed publications, 8 patents, more than 1,700 citations, and has obtained over $7.5 million in competitive research funding. I'm very happy to welcome you, Professor, to this session, and the session is all yours. Thank you.
Thank you. Thanks so much again for the invitation to present. I hope you can see my slides. I think they came up again okay. It's really fantastic to talk to you today about the technology needs in cerebral palsy. It's really illuminating for engineers, I think, to hear about this problem. It's really a lifelong problem. So you can see babies here who, unfortunately, they need to spend the first few days or weeks, sometimes months, in hospital. And at the moment with COVID, sometimes they're often on their own, or their parents are unable to be there at some times. So some of these babies are really at risk of long-term disabilities. Cerebral palsy is one of those disabilities, and you've probably heard of another one, autism. Cerebral palsy is a physical disability, like autism is a mental disability. Cerebral palsy is the most common physical disability that children have that starts very early in life. So here's some pictures here showing you what cerebral palsy can look like. It's really like a brain damage very early in life, perhaps even before birth, during pregnancy. And the types of physical disability, just like in stroke, it depends on the area of the brain that's affected. You're much more likely, everybody is much more likely to have a stroke in their first week of life than even a very unhealthy adult who is diabetic with high blood pressure, hypertensive, and smoking. These babies are more likely to have a stroke. And it's amazing that some babies are able to overcome that. And even if they have a brain injury, they have relatively normal physical functions. And other babies have a very small brain injury, but can have quite dramatic problems with their physical function. They may be in a wheelchair for life, and they may be unable to speak for their life, because obviously when we speak, we're using our voice muscles. Most people that you see with cerebral palsy will have spastic cerebral palsy with the brain injury in the motor cortex, and that will mean that they have trouble releasing their muscles, or that their muscles catch in a spastic way, and they can't perform movements in a fluid way. But there are also some other types of cerebral palsy where the movements are happening all the time, such as the other one shown there. But the big difference is between this disability and others is that it really depends on the injury, but also how people grow up, how they develop, what sort of family life they have at home, and how much therapy they receive. So it is a big problem around the world, cerebral palsy, and we don't even, in some places, we're not even sure how many people have it. It's estimated in India, for example, that it's between 0.5 to 4 people in 1,000 with cerebral palsy, and that's obviously a huge range. And we should be more collect more information to know how much of a problem it really is. It's a video here to show you what some engineers are doing and what cerebral palsy looks like. So the wonderful thing about that research is that it's really led to a product that is now being tested in the US and hopefully soon in Australia and other countries. And that product can help train people at home to improve their walking, but could also be a wearable robot for people to use every day in their lives to be able to, reach the goals that they want to reach and reach the level of function that they want to. Cerebral palsy is really complicated in how it occurs. We really don't understand how all the factors that lead to cerebral palsy. But when I look at this complicated web of causation, I find it easier to think of some of the risk factors I had as a baby. I'm a twin, so I was in this category here of multiple births. And being a twin, I had a low birth weight. I was small for my gestational age. It was a C-section birth, breech presentation. My mother was over 35, and there's other risk factors there. I had jaundice. There's many things here. You can see many of these factors are coming together and increasing the risk of cerebral palsy. So in some of our research, we're trying to collect the data to understand this better and build the models to do that. And I mentioned before that this startup that's looking at resuscitation, which is another big risk factor for cerebral palsy. So it's fantastic to see this type of device being developed and getting out there in the market. So in our research group, we look across the lifespan of what are the technologies that people need and people want. I think that's really the place we need to start at, because in this area, lots of technology, unfortunately, is abandoned. There's an abandonment rate. People stop using wheelchairs and other assistive devices at a rate of up to 70%. So it's really a huge loss. And as engineers, that's obviously something we really need to think about carefully. Is, are we making the technology that people will want to use and keep using? So we're looking at technologies, right in pregnancy or right after birth. There are ongoing trials of different stem cell technologies to treat the brain injury. I'll talk about a bit more about some of the NICU technology, like the resuscitation monitor. We're also making smart toys. So these are some of the pictures down here. So a smart cot mobile that will help the baby perform some physical therapy, and we can also monitor how their physical development is going. Monitoring some of the sounds that they hear maybe in the NICU or early in life, and having mats for them to either crawl on, begin to walk on, or as they get older, maybe if they're interested in esports, they could begin to play games on these dance mats and play computer games to help improve some of their rehabilitation activities. They could wear brain-computer interfaces in a baseball cap like this. This is one of the brain-computer interfaces we're working on with a augmented reality kind of gaming interface. And in here, you can't see too well, but this also has some eye tracking cameras. So we can start to see what their eye movements are doing, hear their voices. Michael Fu will talk again later about some of the hand function projects that are going on. And then as people get older, we're thinking about implanted bionics, how they can interface with robots to help them, and how they would use telehealth platforms like the ones that Nigel was talking about. And I'll also, just like the wearable robotic, I'll also be talking about exoskeletons, which is there's a lot of groups working on that sort of exoskeleton technology for this area. So the research group I work in is heavily clinical and with a lot of what we call allied health professionals like occupational therapists and physical therapists. And they're really looking across the area of cerebral palsy in these ways here. So what is the cause? How can we prevent cerebral palsy? Including a viral infection called cytomegalovirus, CMV, which is something to look at. It's been recognised as a very high risk factor for brain injury in babies, and also having an impact on autism. And some simple preventions like magnesium sulfates and things like that have a big impact and have been able to reduce the rate of cerebral palsy by a third here in Australia. Early treatments like early diagnosis and assistive technology. Some cures like EPO or stem cells to treat the inflammation in the brain, and also some of the policy. So strategies around disability and cerebral palsy, how to include people in education and work. And here in Australia, we have something called the National Disability Insurance Scheme, which is it's unique in the world, and it's like a national health scheme specifically for disability to help get people with disability more included in society. So in our the technology area, we've sort of mapped those to different technologies, and I'll go through each of these in turn and talk about some of the projects listed in the boxes here that we've identified as some of the most important technology needs in cerebral palsy. So I talked about causation, showing you this picture already. We want to include here genetics, the register data we have, which is in Australia, we do track every person with cerebral palsy, so we know how many people there are and how well they're receiving services. And we have access to clinical databases and to do natural language processing and things like that to understand that data as well. We're looking at the causal models, so making a model of this this difficult web of causation, and also importantly including what are the percentage risks on each of these factors, and what are the health economics of actually making a difference and a change. And that's all to get more funding and talk to the government about what we should be doing in this area, because there is a big a big cost to the government and society in this area. So in terms of prevention, medical imaging is obviously very important. Many babies here in Australia will have an MRI or an ultrasound of the brain. And one of our projects is working with a unique data set in the neonatal intensive care unit in one of the biggest hospitals here in Sydney, the biggest hospitals in Australia. Actually, I think it's the biggest hospital in the Southern Hemisphere. And that's looking at this data that's been collected over 10 years now at very high resolution. So that's ECG waveforms at 500 hertz and above. But as well as that, blood pressure, EEG, and the settings of the ventilators, and some of the pharmaceuticals that are delivered to the babies and things like that. And that's been used already to predict things like a blood infection, sepsis, or a bleed in the brain, a hemorrhage in the baby. And now we're moving on to these two conditions, necrotizing encephalitis, which is a gut inflammation, and that leads to poor gut health for the rest of their life, but also can affect the brain as well. And the PDA, which is the part of the heart of the newborn's heart hasn't properly closed, and obviously that causes a cardiac problem. So many of these babies go on to cardiac surgery as well, and they need to overcome that. So using this data, we're sort of building a interface like this, of like a system where we can help the clinician here look at all the data combined into one easy interface for them to make their clinical decision. So it is using artificial intelligence and machine learning, but the clinician is the decision maker. They're completely in the loop, and there's no automatic decisions being made. It's all obviously going through the doctor. In addition to that, there's lots of interest in the pregnancy and other issues in newborn care. So measuring what's happening in low birth weight if babies don't have enough body weight. Preeclampsia is a risk factor. As I mentioned, infection, and there are brain seizures as well. There's the resuscitation monitor, and there's the stem cell therapies that we're also developing technology for as well. So in terms of treatment, as was introduced in my background, there's two areas that we're looking at, which is mobility and communication. In mobility, we've got the robotic walkers that we saw in the video. There's an exoskeleton here from UC Berkeley, which was made for teenagers with cerebral palsy. There's some toys that we're looking at developing, like the ones I mentioned for in the cot, or for babies who are crawling and walking. And there's lots of pharmacology around reducing spasticity to help people move when they're young and keep their movement happening, which helps their bone development, but it also helps keeping them healthy as well. And in terms of speech, we're looking at the eye gaze technology, the small cameras like this one shown here that looks at the person's eyes. They're able to communicate that way if they can't speak. Recognizing their dysarthric speech, if it's hard to understand speech. So typically these people can't use Siri or or Google to do speech recognition because their speech is not recognized, it's too different. And we're working with those companies like Google and Amazon to help make those devices understand these people, and also brain-computer interfaces. So this is me here wearing a brain-computer interface, and my graduate student, actually he's from Bangladesh. He's using a different type of brain-computer interface. And here we're imagining what it would be like to be able to communicate with each other just between these brain-computer interfaces. So at the moment, it's a recording interface. It's not a stimulating interface, but there's a lot of interest in the stimulation as well. In terms of cure, we're thinking about bionics, neural interfaces like the ones that Nigel talked about, but maybe in different areas of the body. So particularly interfacing the nerves that go to our muscles and our pain receptors and our proprioceptive sensors as well. So this could help address mobility and pain. Pain is a big issue for families with cerebral palsy. They often their communication with their children is not great. You know, the child may not have speech, but they know they're in pain and they're not able to communicate that. So that's obviously a big concern for parents, as well as incontinence and having that sense of touch and proprioception to learn the movement when they're a young baby. So we have a project with Greg Swanig, who works with Nigel, and you heard about, putting an electrode array around a peripheral nerve like this. This is actually, components of this are part of the Bionic Eye project, I believe, and that's that's showing the device, the electronics of the device talking to the electrode array that would be around a sciatic nerve. In this project, we're looking at ways of making a better model to test this in, because it's very difficult to test in animals, but it is possible, but very difficult. And we could do that with some tissue engineering. And we can also look at how stem cells are delivered with tissue engineering, and look at embedding some of the neural growth factors in hydrogels, which could be a way to sort of infill the brain injury and give the child back that missing part of the brain. This is obviously quite a thing to think about doing in babies, but I show a picture of a baby here with a cochlear implant to remind everyone that we do that with children, and we do it because we want them to have the sense of hearing early in life, so they can learn. It helps them at school, it helps them learn how to hear, but it also helps with a lot of their other learning, and we need to do the same with touch and mobility. So that's that project in a little bit more detail, just showing you that some children, they receive a surgery in their spinal cord like this, that permanently cuts some of these nerve fibers to release the spasticity, because it causes a lot of pain and a lot of bone deformities. And because that's permanent, they still often end up in a wheelchair, and we want to instead use bionics to have a temporary way of interrupting this feedback loop that's not working well, and provide the right kind of blocking and selective stimulation to enable them to have more normal function. So we're also involved in the technology side of policy, and that includes the regulatory side of things. Software as a medical device regulations. The IEEE has some working groups in this area, which is fantastic. And as I mentioned in the earlier talk, we work with an organisation, part of Cerebral Palsy Alliance, called Remarkable. And anyone here in the call can join Remarkable. They have their startup accelerator is open right now. So if you're interested in this kind of area, you should definitely get in touch, right now. Because they're really welcoming teams from any country in the world. We had a team earlier this year in a Remarkable session from Morocco, and they did really well. Different design problem for different cultures, but we all learn more from that. So I mentioned communication, and I just wanted to now play another video about how communication is really changing for people with cerebral palsy with technology, and it might spark some ideas that you can think about as well.
Can't hear that, Alistair. Why?
You can't hear
shared
that?
it, did you enable sound?
Oh no, you're missing it. Yeah, I mean you can see the text.
Yeah.
Let me reshare. Thanks for letting me know. Yeah, this should be better. It's coming through. And PowerPoint's saying that it's working out a few details, and the slides will appear when they're ready. I will just see if I can find another way to share this video. It looks like it's a little too much.
The video you shared, we couldn't download it from our side, Professor. I mailed you back. That was the issue.
Yeah, I think the let me share a different screen. I think the security of the was too high to be able to share it with you. Let me click the share computer sound. I'll try one more time.
No issues. We can just try.
So this is the video. That's should probably work with the sound. Can you hear the sound now?
So now, job he works at Phillies.
I walk in elevator. I do that.
He has the same hopes and aspirations that most people do. Cerebral palsy is an aspect of his life, but it's certainly not his life.
And I say, ego.
When we lived in the last house, any little mechanical need that he needed to adjust, TV getting water, turning a light on because it was dark, any of these things, that fell to us. He had to call to us to take care of it. We were in and out of that room constantly to adjust things like the volume of the TV, or the channel would change, or the room got dark and we needed to turn lights on, or we needed to adjust the shades on the windows. I think one of the important things that voices have brought to us is a little bit of liberation, not only in the day-to-day, but in terms of adopting things as they come forward.
I don't have to call my mom and dad or my electrician and tell them Hey, I need this, I need that. I can do it independently. I got right on.
So VoiceIt has actually given Nat an opportunity to touch so many more things that are now in the mainstream. This allows him to do the same things other people do and more with his house. And that's what VoiceIt has opened for us.
I need it all the time. I needed to do the technology. I need VoiceIt, do everything.
So hopefully you heard at that time. And you can see that it's a technology that's really made a big difference to Nat's life and to that of his parents as well. You know, it's taken the need, some of the need away from them to be there all the time to do things for him, and it's given him such independence there. You can see that he's able to do things at home, but he's also able to play computer games, and he can also work as well. So it's a relatively seems simple technology, but you can see that hopefully if you could hear his voice, it's really quite hard to understand unless you really concentrate, and then it's easy to make mistakes. So it's great to see that Alexa's now able to understand some of the things that he says. So we had a bit of an influence in that technology happening, and in Amazon deciding to acquire that technology. We ran a technology summit. So this is the Cerebral Palsy Alliance. They're here headquartered in Sydney, but it was their decision that they go to San Francisco, where all the technology companies, many of the large ones are located, and hold a summit there, particularly on assistive technology for cerebral palsy. And these three families came to that summit, along with many engineering groups from a lot of universities in Europe and the US and Australia. And they really presented different problems. Bill Jr. here, he has the ataxic type of cerebral palsy. He has lots of large uncontrolled movements. He's not able to speak either, but he's really interested in brain-computer interfaces, including the type of the UTAR array that Nigel mentioned. His family, they're thinking about that type of technology, and it's really amazing that that BrainGate group and Lee Hochberg, who wrote, he was one of the authors in that Nature paper mentioned before, they're thinking about how they can enable that technology for communication in cerebral palsy. So picking up different signals from the brain and helping people talk and communicate using those signals. Michelle here and Lily, Lily was really having trouble communicating at school. She couldn't get the words out fast enough. I think a lot of people sometimes have that problem, but it's it can be much harder for people with cerebral palsy. If you can imagine, they don't have full control of their voice muscles, so they have that extra delay, and that really causes a problem, of course, at school. When you imagine that, you think that a lot of our testing at school is about how quickly we can answer questions, and a lot of the cognitive testing is like that as well. So there's people with cerebral palsy who have very good cognition. Some of them are lawyers, and they have other really complicated jobs, but they're not able to communicate at the same speed as us. But there's a lot of other people who maybe their kind of true abilities are not realized because of that limitation. And Sophie here, her daughter had just started using eye-gaze devices to communicate. So we heard from those three families, and we talked to these technology companies. They all sent engineers to the summit, and we discussed what technologies could happen in this area. We heard about the VoiceIt, or the TalkIt device that Amazon acquired very recently. And we heard about the BrainGate array that is a brain-computer interface that'll help this lady here drink on her own for the first time. It's about 20 years after having a stroke, so it is a similar situation to cerebral palsy where the injuries happened quite a long time ago. But of course, she had learnt in her life before that how to move her limbs, whereas someone with cerebral palsy will never have had the opportunity to do that. So it is really a new research problem for them. And we learned about this exoskeleton that is from Spain, and it looks like a very engineered prototype, of course. It's very noisy as well, but it has some really fantastic features. The person starts using it with full body weight support. So it's kind of shown here that their feet are completely off the ground. They're not touching the ground. And then as they progress in their therapy, they're given more and more weight to support on their limbs. They use EEG and accelerometer, an IMU on their head to measure their posture, to see if their posture is upright for walking. And the system also is able to identify objects, and so it can be they can be walking around a clinic rather than being on a treadmill, which is the way a lot of the current devices are. And they showed an amazing video of a girl, this girl shown here, who was able to transition from a wheelchair to crutches to walking without crutches by using this device. So it's very encouraging indeed. In Sydney, the key projects we're focusing on are in the area of prevention, the brain cooling, the data analytics that I talked about already, brain cooling, which is a very important technology and something that would be completely appropriate, I think, in India as a method for reducing brain injury at birth, and the resuscitation monitor I showed as well. In children, we're with a group in Hong Kong, we're developing a soft exoskeleton for children under 2 years, so none of those sort of hard mechanical motors or metallic parts you saw in the previous exoskeleton. The exoskeleton we're working on is just a soft pneumatic devices, like a soft balloon that is under the knee and is inflated to help the child begin to stand and step and walk. And we're working on the neural interface project and the brain-computer interface with the BrainGate group for looking at restoring lots of abilities. So when I talk to people with disability, spinal cord injury, cerebral palsy, lots of different disabilities, they all have different views, of course, about what sort of technology they want. It's a bit like having, our iPhone or our computer. We turn on different settings. We either use it like a super user, with low-level code, or sometimes we just like to leave things with the most basic computer interface. And so this needs to be the same, I think, in the technology that we look at. And it's great to be working with the Cerebral Palsy Alliance, who are willing to support projects at all different levels like this for different people. So I'm typically asking people now, would in the future, in the year 2040, when we have all this technology, you may be in a situation where you can choose. Would you like a robot to be helping you to transition from your bed to your wheelchair? Would you like to wear a robot, like a wearable robot or an exoskeleton, like shown here? Or would you like to be the robot and have a bionic implant? Is that something that interests you? So I think I'll finish there. It's great to talk to you again, and thank you for the invitation.
Thank you, Professor. Thank you, Professor. That was a wonderful talk, and it's very heartwarming to look at how technology can help brighten the lives of people who are really in need of humanitarian support. And your last question was really interesting. Would you need a robo to assist, or wear a robo, or be a robo itself? So that lays the road ahead for future research and innovations, Professor. Thank you. Professor, one basic question I had, like, what is the difference between a brain-computer interface and a neural interface, Professor?
Well, I think both, a brain-computer interface could be called a neural interface for sure. But we're thinking about also the interfaces to neurons that are not part of the brain. So these would be maybe the neurons in the spinal cord, but in particular the neurons that are leaving the spinal cord and going to our muscles or other other parts of the body. So like a sciatic nerve would be a great target because it's a nice large bundle of nerves.
Okay. Thank you, Professor. And Professor, the cerebral palsy, how earlier could it be detected, Professor?
That's a fantastic question. It's thought, at the moment, the general idea is around 3 years of age, or maybe even 5 years of age or older. But the goals are now to recognise people who are at least at risk of cerebral palsy at 3 months or earlier, to give them as much intervention as their parents would like. Really, we would like to give them as much therapy as possible, and that they can afford. But and I'd like to mention there's a really interesting project where you can now record your infants on a mobile phone, record their movements with the mobile phone camera, and you can send that video to be assessed for cerebral palsy.
Well, that's nice to know, Professor. Thank you for your answers. Any questions from the audience, the participants? Professor, in one of the slides you mentioned about in the sepsis condition in NICU. So I have actually learned about the sepsis condition for adults in ICU. So how does it affect the babies also, like sepsis condition? So could you throw some light on that?
Well, it's very dangerous. Many babies die of sepsis.
Okay.
Unfortunately, I have a new student from Bangladesh who's working on brain-computer interfaces. He's very talented, but unfortunately, his daughter, was in the NICU for a long time. And when you're in the in ICU for a long time, you're at risk of infections like that. One of the common tools is to measure changes in heart rate variability that are a strong indicator of sepsis, or obviously other conditions as well.
Okay. Thank you, Professor. Thank you. Any more questions?
Yeah, I have a question. The devices, Professor, you were talking about in Amazon, do they have any standard devices to interface with before getting connected, or they are actually independent and they can work on their own? The Talkit. Do we need? Yeah. Do we need?
Device?
Yeah.
Yeah. No, that works. That's a software solution. So it is a piece of software that sits, can be in a tablet, in a phone, and it sits in between the microphone and Alexa. So they're able to recognize the dysarthric voice, and then tell Alexa or whatever the system it's using what the actual command is.
Okay, so you can connect it with any device, any device. That's wonderful.
Any Amazon device at the moment. That is a really good point. We have Google and other companies contacting us at the Cerebral Palsy Alliance because we have a large group of people you know, cerebral palsy interested in research, and they would like to record their voices so they can train their databases. But of course, it's very difficult for these people to communicate. So if they need to talk, you know, for Apple, for Google, for Amazon, it's a lot of effort. And one of our projects is to develop a database that then we can share with each company. And that would reduce the burden for the people with the disability. They would benefit.
That's a great support to those people. Wonderful, Professor. Thank you, Professor.
Thank you. Thanks for the great question.
Participants can unmute your mic and raise your questions.
Okay, I think, ma'am.
Madam?
This session we can close.
Yes, madam. Yes. On behalf of IEEE EMBS Madras section and KSR College of Engineering and Technology, I thank Professor Alistair McQueen for the session on technology innovations for cerebral palsy. That was a wonderful talk. Thank you, Professor.
Thank you. It was a pleasure.
Thank you.
Thank you, Professor. Thank you for your time and
Thank
being
you.
with us today. So the next session we begin. Now it's Professor Rifai Chai. I would like to welcome Dr. Rifai Chai, Academic Director, Pathways and Partnerships, Senior Lecturer, Department of Engineering Technologies, School of Science, Computing and Engineering Technologies, Swinburne University of Technology. So Dr. Rifai Chai received the B Engineering degree from Kridawakana Christian University, Jakarta, Indonesia in 2000 and the PhD degree in Biomedical Engineering from the University of Technology, Sydney, Australia in 2014. From 2000 to 2011, he has worked as product development engineer with companies in Indonesia and Australia. Currently, he is working as academic director and senior lecturer with School of Science, Computing and Engineering Technologies, Swinburne University of Technology, Melbourne, Australia. His expertise, teaching, experiences and research interests are software, firmware, embedded systems, artificial intelligence and machine learning, brain-computer interfaces, medical technologies and autonomous system. His research projects are medical device, assistive technology, smart wheelchair, driving related research, mental fatigue classification, rehabilitation, biomedical modeling, AI and advanced computational intelligence and embedded systems. We welcome you for the keynote address on brain-computer interface with artificial intelligence. Happy to have you with us here, Dr. Rifai Chai. The session is all yours. Thank you.
Thank you very much, Professor Jayanti, and good afternoon or good day to everyone. I noticed there's a few colleagues from Australia as well, so it's good to see a lot of people from Australia, and also some invited speaker from around the world. I see that. And once again, thank you very much, Professor Jayanti. And let me share my screen. Sorry, I think I can't see something from that point. All right, tell me whether you can see the screen. Are you able to see the presentation screen on your side?
Yes, sir.
Yes,
Oh, that's
Professor.
all
Yes.
right. Yep. Today my talk is also related with brain-computer interface, so it would be similar talk to Professor Alistair as well from University of Sydney, and in different application of brain-computer interface. So before I start with the particular project, I'll try to introduce about the topic first. So we just all these things is back to the Industrial Revolution. If we understand that, you can see we have the start from the pre-industrial revolution when we harvest the energy from the animal. So when we started to have a water and steam engine, that what we call the Industrial Revolution 1.0, and the next one is when they come up with the electrical or electric, they come to the Industrial 2.0. And 3.0, you can see here when they start to have the computer coming in at the end of the 20th century, so 1990s. So when they have the computer revolution, they have the IBM, Microsoft, and so on. That's become a booming of Industrial Revolution 3.0. And now we are in the era of Industrial 4.0. So in that era, what we call a cyber physical system. So starting to have more with the AI there as well, and some of the human interface. But in the future, that's the guessing is in the 5.0 is we almost there in the 5.0, which is will be a fully AI and also something to do with the human machine interface, which is related to my talk. Well, part of it with brain computer interface. And just to give you the idea again, brain computer interface or BCI, basically we just want to bypass the whole things, just read from the brain signal. We measure the intention of the persons, and after that use that one for control and communications. And there's a lot of definition they keep updating about the definition of BCI, but this is one of them. And you can see some of the technology of BCI can be invasive and non-invasive. So give you a little bit review. So on invasive, mainly on using electrical signal, and they can be just using the micro electrodes, put it inside the brain. So it's invasive. That's why they measure it straight away, use the electrical signal from the brain. Or can be a semi-invasive, what they call a ECOG, so electrocardiogram. And on the non-invasive side, which is the one that I'm normally using in my research, which is from the electrical, and a lot of people use that one because non-invasive through that electroencephalogram EEG. But we can measure as well using the magnetoencephalography MEG, and also some of the imaging technique, for example, FNIRS using the near infrared, or fMRI, PET. So it's pretty big machine that you can find it in a hospital. Now, in terms of the publication, this is the publication. You can see so far a lot of concentration using the EEG around the world. I use the keyword BCI only, but there's a lot of term like Professor Arisa mentioned about Neuralink as well. That's linked to the BCI, but I just use a BCI term when I do these searchings. You can see EEG is quite a hot topic around the world in terms of the publications. And the other one is the fMRI. This is neurotechnology, neuroscience. A lot of people with the fMRI as well when they are doing the research. And some of them the invasive. Mainly for the invasive, like the brain get and sort of thing, they concentrate on America, but with the collaboration, they can combine with around the world as well. So this is in terms of publication. You can see started from 1990s when the era of the Industrial Revolution 3.0, like maybe less than 10 up to now. So 2000, because we're still in 2021, we haven't finished. So 2020 is around that 800 and above, 900, almost at 900. You can see that that publication is they become more and more until now from the beginning of 1990s. And in terms of the BCI and EEG method to use for the application, if you see the state of the art of the research, it basically can be divided into two. So they can use a selective attention, which is need a certain stimulus. So one of them is called P300, called peak after 300 milliseconds. We can see that features is P in the brain. Or some of them they use what they call a steady state of evoked potential. So if the stimulus on certain frequency, that frequency we can detect that one as well. And I use actually SS3P one of this one for one of my application, and also the other task what they call a spontaneous signal. So spontaneous signal means it's spontaneous from the brain. So you just one of them is popular, which is the motor imagery. So if you search a publication, a lot of paper that discuss a pretty hot topic on that motor imagery. It means that you just imagine moving your hand, right hand versus left hand. It means you can detect that one as well. You can detect the features. And previously they come up the initial with what they call a slow cortical potential, but to make it real time is pretty hard in terms of the BCI application. But probably in terms of the rehabilitation, we can use that one as well. It will depend on the particular application of the BCI. And the one that I present in here is more related with non-motor imagery, which is the other mental task that we can use as well for the BCI. And in terms of the applications, so basically we can divide it into three. So active, reactive, or passive. So active is the user is actively performing that task or mental task. So whether it's a motor imagery or non-motor imagery of the mental task is one of the category to the active BCI. Reactive, it means that you provide the reaction of the particular external stimulus, for example, the P300 or SS3P type of the BCI. So that's categorized into the reactive BCI. And another point of BCI, these days, sort of when you perform a task without voluntary control, and some of the group we call it a BCI as well. It's not active or it's not reactive. It's passively, for example, monitoring the brain wave, like we're doing in a mental fatigue, is what can be categorized into a passive BCI. Okay. And the topic I've discussed is not just about the BCI. This day, BCI need to do with the algorithm. So what we call an AI. That's why the topic of today's discussion is combination of the BCI and the AI. And this is a general topic. So you can see that measurement of the EEG. So mainly I'm using EEG in my research. And the rest is the signal processing, the features, and the machine learning. That's part of the artificial intelligence, or what we can call it a computation intelligence. And the rest is the output used for certain application. And I'm going to present to the topic. I think that depends on the time. Hopefully I can finish this one. So I'm going to go through mainly not that detail in terms of the method, but probably you see some of the formula there, but I'm not going that further detail, but you can have a look in the publication later on. So one is for brain control wheelchair. The second one is mental fatigue detection or classifications. And this is few of the main publications, journal or the conference. They got some more. You can have a look into my research or with the collaborator publication there as well. And brain-computer interface or brain control wheelchair is quite important for people have the disability. You can see for alternative hands-free control, there are few technology we can find. And from the sip and path, voice control, muscle control, head movement. So it will depend on the disability of which area. So on certain condition, what they call it as a lock-in syndrome. So it means the brain still functioning. For example, the amyotrophic lateral sclerosis, the brainstem stroke, the brain still function, but the other they can't move at all. So they can't move their hand, they can't move their leg, but the brand still okay. So that's the reason we can use that the BCI as an option to they can use it as a control. In this case, it's control wheelchair. And at that time, we conducted a study with healthy and also people with a high level of spinal cord injury around that cervical area. You can see C3, C4, C5. So we've done that study in the past. So we've got the ethical approval, and we mainly concentrate on non-motor imagery. Okay, we want to see the other different mental tasks, whether we can use that one to control the wheelchair. So this is the task that we use. So just by imagining or letter composing, arithmetic, okay, imagine the rubric keep rolling in the head. So also that some additional tasks related to the eye closed and eye open, because we know that's the EEG using the alpha eye closed. We know the features is quite dominant around that what we call an alpha band, which is the 8 to 13 hertz. That's why we including that one as well. We can use that one for additional control for the wheelchair in real time. Okay, when we talk about our features, so we try to explore different features that we're going to use. So when we talk about AI, it's not just only the machine learning, also we can go into features extraction as well. And we found it, normally in EEG, people will use the frequency-wise features extraction. But I tried to use other features, which is I found it using the Hilbert-Hwang transform, which is combination frequency and time domain analysis that can give a better classification that we can use. So this is only the theory of the Hilbert-Hwang transform. Yes, you can have a look in more detail of this one. So this is how we program the Hilbert-Hwang transform. That's more about the algorithm. And because we're recording the eye closed signal, that's why we check whether our Hilbert-Hwang transform, the eye closed signal is giving is showing in the domain area when we apply the Hilbert-Hwang transform. So you can see on the right hand side here, which is after we apply the Hilbert-Hwang transform, which is the spectrum, we can still see the alpha wave on that region. So then we talk about the classification and algorithm. So at that time we develop our optimized version. So the classical one is the structure is what we call artificial neural network, but we propose at that time a new optimization. This is combination of particle swarm with a fuzzy and optimize the pure parameter in the particle swarm optimizations. And we do a comparison as well for different machine learning that we know so far. And this is just about the algorithm. And this is about how we prepare the data of the EEG. So you can have a look in more detail in the paper. And so because we collected data, we also collect data using OSISOM, but later on I'll talk about the hardware that we developed as well. And this one is the result of the classification for the eye closed and eye open. And we need to make sure, because we use different features, and we need to check whether the alpha eye closed using the features extraction and the AI still provide a good result in some of the eye closed as well. You can see the accuracy is above 90 or nearly 100%. So I mean the machine learning and the features extraction is working, the one that we propose. And we do a comparison as well. This is the mental task classification. And this is 3 mental tasks, because we're going to use that one to control the wheelchair using the AI that we propose. And we just want to check, in different time. But later on in the wheelchair, of course, the fast is better. But you can see in the research, we're comparing actually what is the best time window that we can use to control a wheelchair. You can see from comparison as well, different algorithm. So you can see the one that we propose is much better on the other machine learning but also the different time is getting much better as well. And this one is comparison with people with disability and with normal normal people. And you can see it's a 1 second time frame windows is quite different, but when we increase the window, is actually converge become similar accuracy at the end. So it means that people with disability need more time, which makes sense, yeah, in terms of the data that we got it, in terms of the result as well. And this is the result that we got it in terms of the proposed method. Different AI, so this is the one that we propose, and also different features extractions. So at the end, we can up to around 80 something percent of the accuracy for the 3 mental task classification for this project. And this is only for the purpose of the publication. We try to check the statistical significance, and we put it there is all of them is significant compared to the other method. And because we want to do real time, so in here we just wanna, because the EEG that we get is more channel. It's around 6 or 7 channel, 8 channel there, or 6 channel. But we want to reduce because of the practical, to make it more practical. Less channel is better. So that's why we select different, the optimum channel that we can use for real time. But we just want to make sure it's optimum, is comparable at least to the complete channel, 6 channel that we get previously. You can see the accuracy is comparable around the 80%, and we use that second channel in order to do a real time later on. And this is the one that I mentioned. We also involved with designing the hardware as well, because at the end we think about the real time, the product, and on the latest stage as well. Firstly, we're just using the conventional gel-based EEG. We'd also developed non-gel based as well for this system. And this is the specification of the EEG that we develop. So we just need to make sure is good EEG is comparable to at least comparable to the medical grade of EEG. So this is the whole system. And this is the EEG, and this is the embedded system. We use another embedded system to control the computer, basically. And this is testing the system using the EEG that we developed. So we have to make sure the alpha I-class is there. So you can see here, there's quite a good signal of the alpha I-class, with the hardware that we developed. And this video is showing you a real-time control wheelchair with the proposed method with the less channel, 2-channel EEG. The white stuff there is the EEG that I wore with a headband. It's 2-channel only, so we can actually control the wheelchair as a BCI control wheelchair project. Okay. This is slow, but we can see as well from the result. We can use 1 time frame window, but with less accuracy. But we increasing more time, that will become much better accuracy. But that's how we implement in the real time as well. I think in wheelchair here, I just use around 2 to 3 second time window to perform in this real time. Okay. I just stopped that one so we can move quickly to the other project. So the next one is the brain control wheelchair is active BCI. The second BCI that I'm going to present here is about the passive BCI. Is one of them using the mental task. The application, one of the applications we can use that one to detect what we call a driver fatigue. And this is a few of the publication related to this project. And the idea is fatigue in transportation is very important, because and it's dangerous when people having a fatigue in transportation. You can see in the statistical that we can find in Australia, New South Wales and Victoria, and it's become a top of the casualty crash related to that fatigue issue there as well. And For the measurement, there's a different way to measure the fatigue, you can see, but the concentration for us is the measuring using the brand technology, so using the non-invasive, which is the EEG. So this is the signal of the EEG, and this one we actually collaborate with University of Sydney, with one of the professors, Professor Ashley from Sydney University. And this is, so they have collected the data, 32 channel, and doing that driver fatigue in a simulation. And we use the AI to deal with the classification of the driver fatigue. So you can see the signal processing is pretty much same that we have discussed before, but there's a features extraction there as well. And this is the frequency-wise features extraction that normally used for EEG analysis. But in one of the paper here, I introduced auto-regressive modeling method, because I found out that one as well is, we can use that one to improve the classification instead of using the FFT or normal power spectral density method. So that's why one of the paper actually used the AR method as the features extraction. And the classification, so we use the standard Bayesian neural networks. So this is have been used very popular as well around that machine learning people. And one of the paper actually used deep learning. So I modified slightly the deep leaf network, so it becomes sparse deep leaf network. So we published one journal using this AI for the driver fatigue classification. So basically, it's quite different, the structure of the deep learning compared to a conventional neural network. So conventional neural network maybe have the only discriminative part only, but you can see deep learning, they have also the generative layer, and also they have the discriminative layer. So combined together, it will be part of the deep learning structure. It's quite interesting when we do with the deep learning, actually it can be improved a lot for the data. So you can see this is the optimal structure of the machine learning that we use to make sure we get the optimal structure first before we apply it, before we using a testing set. And this is the information that you can find more detail in one of our journal. This is the test on the different layer, because we applied deep learning, but actually we in our deep learning, the 2 layers is more than enough for this project. Actually, we don't have to go with a much more higher layer, because actually it's downgrade the performance. But deep learning, but the nodes is big compared to the standard neural network. And this is the result that we have it in a journal publication. You can see using the standard power spectral density is around 80-something% using the one that we propose, and also the another features extraction method, which is auto-regression method, we can actually increase up to 90-something%. And this is the statistical significance. But that's what happened at the moment. So I have the simulator at Swinburne as well. So we try to implement the real time in a simulated version for the driver fatigue classification, and also different driving through the mental different driving related mental, for example, the distracted driving as well. We're gonna have a look at detail in few of my research student doing that a different project related to the driving. And also one of the research student, also another research that we start doing at the moment to apply that mental fatigue or mental workload on the aviation as well. So hopefully in the future I can present more about that research. I think I checked the time. I think so far that's it from myself. Hopefully we can go with the question and answer. I'm happy to answer the questions. Okay, thank you.
Thank you, Dr. Rifai, for your presentations, both the presentations. And any questions from the participants?
Participants can unmute your mic and raise your questions.
Professor Rifai, thank you for your presentation. I have I have a query. Like when you were mentioning about certain time windows for EEG measurement, 1 second, 2 seconds, and the ideal time that you found from your research was 7 seconds. I was intrigued by how in 1 second can we understand what a person is about to think, like turning a wheelchair right or left. Is 1 second time window is real? In reality, is it true?
So we're talking about the EEG response. So when you measure, so when you cross, of course, with mental tasks we need more time, but actually it's a response time of the EEG. When we go into a design EEG, normally EEG we have the 256 Hz. So it means that in 1 second we can actually get the EEG data. So when you close your eyes, that actually is what happened in your brain is actually we can detect that one very soon as well. But on the mental task, that's why we need more time actually to doing that one. So you can see that the accuracy that one is affecting. But in terms of the EEG classification, so we can. That's why we doing that study. 1 second, 2 seconds, 3 seconds. But in 1 second a time, actually we can do a classification.
Okay.
Yep.
Thank you, Professor Rifai. Professor Rifai, you had selected certain channels for mental task and for the eyes closed. So how do we decide on which channels are optimal for any task? For example, for moving a wheelchair, for mental task. So is it based on some literature study or there is some standard way of knowing?
Yep. Depend on the BCI, the one that we use as a mental task. If you're going to do a motor imagery, we know that motor imagery around that central area. So it means the channel that we pick should be on the central area. But because the one that I'm using for control wheelchair is non-motor imagery, so it's a mental task. It's actually something to do. Most of it, we try to avoid the frontal part because it becomes very noisy. It's hard to clean. We try to pick area around at the back, so around the occipital, and then around the parietal, and maybe sometime maybe can say central as well. So you can see in the result or in the journal as well that we publish, we try to see the combination of the two channel, which one is the best that we can use in terms of the result. Yep.
Okay. Thank you, Professor Rifai.
No problem.
Got the answers. Thank you.
And I have a question from my side, Dr. Rifai. Is the wheelchair design commercialized and has it come out?
So the wheelchair at the moment, so actually this is a project with Professor Hung previously and still ongoing project. It's still on a research. We try to commercial that one at the moment. So still in the research, that's why we keep doing that research at the moment.
You are trying to add more features to it?
Then again?
Is it that you are trying to add more features to that?
No, and actually, it's commercialization is a different perspective on research. So we need to find more investor or funding. So, but the research is ongoing research. Yep.
Yeah. Thank you, Professor. Any questions from any more questions from the audience? Participants? Yeah. So I thank Dr. Rifai Chai for your time on behalf of IEEE EMBS Madras section and KS Rangasamy College of Technology for accepting our invitation and delivering this talk to us. Thank you very much, Dr. Rifai Chai. Thanks a lot.
Thank you very much as well, Professor Jayanti.
Pleasure is ours. Thank you. Now, the next session is by Professor Celia Shanas. I welcome you, ma'am. And I would like to invite Professor Thamissarthi to introduce the guest for this particular session.
I welcome the speakers Julia Sanas, Professor, EEE BUET, Chair, IEEE Signal Processing Society Women in Signal Processing Committee for this international collegium. Julia Sanas, a fellow IEB, received PhD degree from Concordia University, Canada, and is currently a professor at the Department of EEE BUET, Bangladesh since 2015. She has published more than 150 international journal and conference papers, and she is a recipient of the Canadian Commonwealth Scholarship and Bangladesh Academy of Science Gold Medal for her contribution in science and technology. Her research interests include the areas of signal processing for speech analysis and enhancement, audiovisual recognition for biometric security, control system, robotics, pattern recognition, machine learning and deep learning, etc. Recently, her papers have received Best Paper Awards in IEEE DissertCon 2019, Biomedical Engineering Track at TenCon 2017, IEEE Wacon ECE 2016, Humanitarian Challenge Track at Autumn HTC 2017, and the Best Interactive Poster Award at ICI DCR 2017. Her paper has been selected for the top 10 Best Paper Awards in the Student Paper Contest of 2018 and 2014 IEEE International Midwest Symposium on Circuits and Systems, College Station, Texas, USA, Knoxville. She was the winner of the Best Student Paper Award 2008 IEEE International Conference on Neural Networks and Signal Processing, Hengqiang, China. She was selected as one of the finalists of the Student Research Presentation Competition in 2009, Cytocom Workshop, Montreal, QC, Canada. She is the recipient of 2021 Inspiring Women in Academia Award from Bangladesh Brand Forum, 2019 Art and Humanitarian Activities Outstanding Volunteer Award, 2016 MGA Leadership Award, 2015 WIE Inspiring Member Award, 2013 Art and WIE Professional Volunteer Award. She has been an IEEE office bearer in various positions and societies. She is the founder and chair, IEEE SPS IAS Bangladesh Chapter, co-founder and vice chair, IEEE RAS SSIT Bangladesh Chapter, founder and advisor WIE AG IEEE Bangladesh Section. She has served as the general chair of IEEE Symposium of Region 10. She has served as an editorial board member, IET Signal Processing from 2018 to till date, and 2018 to 20, a member of Technical Committee, Image, Video and Multimedia, Asia Pacific Signal and Information Processing Association. She has more than 20 years of experience in leading impactful technical, professional, educational, industrial, women empowerment and humanitarian technology, power and energy related projects at national and international level. I welcome you, ma'am, for your keynote address on this international collegium on the topic of computer-aided automatic disease detection from biosignals based on deep neural network. I welcome you, ma'am. Now the session is yours, ma'am.
Thank you. Thank you very much for such a humble introduction. I really am proud of you, Jayanti, and your team. Please, for organizing such colloquium. So let me share my screen very quickly and hope you can see it, right? Dr. Jayanti?
Yes, Professor, we are able to see.
Yes. I really want to congratulate and thank the TNBS Madras chapter and the university who is in collaboration to organize such a colloquium in such a timely topic during the pandemic for timely dissemination of knowledge. So actually, I like to talk about the emerging techniques, and that's why I have chosen such topics so that we can cover interdisciplinary researchers actually. And first of all, I like to thank every single volunteer who is behind it, starting from this poster designer who has played a great role of spreading this information, and so that we can see so many participants today. So we have to really recognize every single volunteer in it. So this is brief bio, what has been just introduced. I really like to invite you humbly to visit my Google Scholar citation, not to know about the citation only, to read the papers and to let me know how I can collaborate with you and how I can help you. So in 2020, when the pandemic started, I was very worried that despite limitations of computational facilities and students are in the village home, and how I will continue my research. At the end of 2020, I found that it has surpassed the citations in 2019. So this is a clear attestation that what my students have done, how emerging topics they have selected, and we tried our level best to publish in impactful journal and conferences. Not only that, the people have found it useful. That's why people have cited it. This is just a message to the younger generation, because I served in a highest ranked engineering university in the country, and where majority of my students are going to PhD in the United States, Canada, and Australia. And I have limitations of students, computational facilities, and funding opportunities. So what can be done in this limited situation? So we can learn from each other, we can inspire for each other. Please go through my Google Scholars profile and let me know how we can come together to empower ourselves. This picture is very powerful. I believe in this power of networking. These are the two human chains that was led by me. And in the left side, these are the technical society chapters and affinity groups, starting from Women in Engineering, and of course, Engineering, Medicine, Biology Society, Signal Processing Society, and other relevant society where I'm involved in terms of my research and professional activity. The right side, for my every research, I try to connect with billions of people. That means I try to meet up the goals that is set by United Nations. UN has 17 SDGs. It is very important for this subcontinent to connect our research to the UN SDGs. Let me share the research. Abnormality detection in musculoskeletal radiographs. So why musculoskeletal radiographs? Because 1.7 billion people are suffering from these abnormalities. And we wanted to automate this detection process to accelerate the diagnosis. And for this, my students have developed a new neural network that is called capsule network, and why? Because there are limitations of conventional neural network. One is poor translational invariance and lack of information about the orientation, and also it needs huge data set for training. So how CapsNet overcomes these problems? Because here the output from each neuron is replaced by a vector, and max pooling operation is replaced by more effective routing by agreement algorithm. So this is a brief flow diagram of this capsule network for the abnormality detection. And that means we will take input images, pre-processing, then convolutional layer optimization, and class capsule determines the output. So most at the very beginning, we need pre-processing because our data set contains image of variable sizes, 64 cross 64, 128 cross 128 and 224 cross 224. If you take a visual observation, you can see in the third case, the features are more included, and the first case, it is blurred. So we depend on the objective measures also, such as training and validation accuracy, and we found that for 224 plus 224, both the accuracy are higher. Not only that, we have evaluated BRISC score and NIC score. The lower the score, the better the quality is preserved. We can see for 224 plus 224, both BRIC and NIC scores are lower. That means more features are preserved even after resizing. That has given us a rationale that we will move forward with 224 plus 224. This is a proposed structure of the CapsNet. Convolutional layer we have, convolutional capsule layer, and finally the capsule layer. This is the structure we have proposed, and this is a detailed scenario of the proposed structure. You can see different convolutional layer, finally the routing by agreement algorithm to really tell whether it is normal or abnormal, and you can see the sizes, how it varies. You know, question is which data set you have chosen or you have utilized. We have done the Mura data set that contains a very big data, 14,656 images. Among them, 8,941 images are normal and 5,715 images are abnormal. So we don't have data imbalance. That's a good thing for us. Now, I will let you know that what is a routing number, how routing number creates impact on our performance. X-axis is the number of routing. Y-axis is the training and validation accuracy represented by black and red color. You know, for all types of images such as finger, elbow, hand, humerus, forearm, shoulder, wrist, we can see the training and validation accuracy increases with number of routing. But in some cases, E and F, you know, for forearm and shoulder, that you can see overfitting occurs. To avoid overfitting, we have decided that we will go for routing. Second question, how the proposed capsule network is effective. We can see that for a conventional DenseNet, our kappa static score is 70.5%. And for our proposed CapsNet, that average kappa score is 80.115%. And if you see this bar graph, you can see for different types of images, the kappa score is 10% better than the conventional DenseNet. Using 50% less training data, that's a very great achievement. Now, question is how you have evaluated your performance. We did by the parameter training accuracy and testing accuracy, and compared it with the state-of-the-art techniques. Compared to the state-of-the-art techniques for different images for the same data set for CapsNet, we are providing better training and testing accuracies. Finally, what is the loss accuracy graph? Our accuracy represented by red and loss represented by black, and for DenseNet accuracy represented by blue and loss given by purple. We can see for every iteration our accuracy is higher with lower loss, and their accuracy is higher with greater loss. So that's a very good example of the efficacy, effectiveness of our proposed structure for the problem at hand. And this has been published by my undergraduate students along with my PhD supervisor. It's a work of 3 generations, and we published in IEEE Access. And it's an outcome of an undergraduate thesis. That's the example. And I was doing, along with my students, to detect tuberculosis from chest radiographs, And, it's an extreme image, black and white extreme image. We have taken the decision from DenseNet, RasNet, and Inception V3, and we have taken an ensemble of decision. And then we have also taken the machine learning ensembles, and then we have combined the both ensembles to have a better prediction. This has been also published by the same group of researchers and has been published recently at IEEE Access. And I was also doing pneumonia detection from X-ray images, but that time that COVID came and my students became more interested with handling COVID using X-ray data and using comparing different data set, different multimodal images like ultrasound images, like CT images, like X-ray images, and to find which is the best imaging technique. And recently, using these multimodal images, they have published a paper on IEEE transaction on artificial intelligence. I really recommend you to read that paper and let me know how we can do more collaboration and develop a better structure to really find it. I'm a lifelong advocate for women empowerment through research and innovation, and I have visited a lot of rural areas of Bangladesh, India, Sri Lanka, Nepal, Thailand, Malaysia, Indonesia, and many other places in region 10 and beyond region 10, and I found that our women are suffering from skin cancer, but they are reluctant to do it because usually doctors see those color images and try to diagnose it. These are colored. Classify what type of skin cancer it is. That will really make the interaction with the doctor easier, and decision making will be accurate and faster. With that motivation, we have started using where the problem is not 2 class, problem is 7 class. But there is another problem, that is in one class there are less number of images, and in other class there are high number of images. That means we have a data imbalance. So for data imbalance, what we did, we have proposed data augmentation using data augmentation library, and we have applied few fine-tuning strategies. That if the large data set and similar to the pre-trained models data set, you can train some layers and leave others frozen. If you have a large data set but different from the pre-trained models data set, you can train them entire model like this. And these are the models we have really investigated and applied: Inception V3, VGG 16, VGG 19, RasNet 50. We have also implemented many other conventional features for the skin cancer diagnosis. This is a typical model architecture for Inception V3. We have convolutional layer, we have average pool, max pool, concatenational layer, dropout layer, fully connected layer, and softmax layer, and you can see the sizes. This is the top layer specification for all models, and we have used all conventional features as follows, like color feature, image quality feature, histogram analysis in color planes. You know, how we have evaluated our result. We have evaluated our results in terms of accuracy, and as expected, compared to the conventional features, our models are performing better. As we have done in case of our tuberculosis, we want to identify the best layer, that we have to take the ensemble of layers in the session and send it to the classifier for a better prediction. I hope it will open up a new direction for improved accuracy for the skin cancer research. Since I'm serving as chair of Women Signal Processing Society, Women in Signal Processing Committee, let me introduce with you the Signal Processing Cup competition and Video Image Processing Cup competition. I have been participating in those competitions since 2015. And my team was ranked 5th in 2015. In SP Cup 2020, team was ranked 4th, and VIP Cup 2020 team was ranked 5th. It's not a matter of ranking. It's important to know the topic to improve your technical skill. You see the topic, unsupervised abnormality detection by using intelligent and heterogeneous autonomous system. You know, although it was done, this topic, but this topic has opened up a new opportunity for my group to employ that research to deploy medicine for the COVID-19 patient, and artificial intelligence and deep learning plays a big role here. And real-time vehicle detection using tracking at junction using a fisheye camera. This was the problem, but this has opened up a new avenue for me for designing a tracking system in the hospitals, in the offices, okay? And which is very important for maintaining health or distances in the post-COVID era. So in 2015, when my team was ranked 5th, and it was a problem related to PPG signals, and they were very upset, but from that topic, we published 3 journal papers, and that I really encourage you to read those journal papers. These are very contributory, and and that has helped my students to get admission in University of California, Riverside, and also Princeton University. You know, it's Ivy League university. So it's important to embrace those techniques to really deploy in the biomedical healthcare domain, which is interdisciplinary in nature. And let me tell you, in IEEE TENSUM, which is the regional flagship conference, I was the general chair for it. We have handled more than 1,000 papers. What I wanted to mention, that it is the first Region 10 flagship conference which was fully virtual. And I want to tell you that people have a lot of interest, and we have a lot of papers in the biomedical and healthcare domain, and a lot of papers who are using this deep learning techniques. Actually, deep learning is a tool, and that can be applied in speech, image, video, ECG signal, EEG signal, EMG signal, MRI, CT, ultrasound image, also in power signals. You can also apply the deep learning for automatic arrhythmia detection, one from ECG signals, to help the doctors to really find the arrhythmia very quickly to avoid fatal deaths. And also another research group are working to apply the deep learning for brain signal, EEG signal, to really detect that whether the patient has seizure, and if seizure, that what is the location of seizure, so that the mitigation plan can be taken. The paper on seizure has published in 2021 in Elsevier, Biomedical Signal Processing and Control, and it is also available in my Google Scholar profile. And I really tell you, request you to read those papers so that we can really come up with new ideas to foster collaboration. Also, in the TENSIM, Professor Toshi Fukuda, IEEE President, was the chief guest, and for International COVID-19 Congress, we had ideas. We have a EMBS colloquium also. We wanted to know how our EMBS researchers, our signal processing researchers, our industry experts, our students, our women in engineers, our young professionals are thinking to really handle COVID. I have found that many of them are applying this deep neural network to handle different COVID-related problems. And also, Toshio Fukuda was also chief guest in COVID-19 Congress. Recently, we did International Women in Engineering COVID-19 Congress, where I found there are a lot of researchers who are applying deep learning and AI to handle COVID-19 related problem. It was really fascinating that how they are doing it. These are my list of award. Actually, it's not a list of award. Actually, it's a list of commitment and hard work. These are list of group awards. Women in Engineering group, for which I was the founder, become the best one in the region 10 and become the best in the whole world, because they really launch many technical activities and conferences, especially the first Women in Engineering conference in the area of electrical and computer engineering in 2015, and then in 2016 we collaborated with Pune section, and 2017 with Uttar Pradesh section, 2018 with Thailand section, 19 with Bangalore section, and 2020 in Bhubaneswar section. So it's a huge collaboration. It has really fostered and inspired many women to come forward to present their research. As a first female chair, I had the honor to receive this 25-year celebration banner from Professor Cookzine, then Region 10 Director, and that has helped me and that has inspired me to launch many new initiatives for my section, and I helped my section to be in 2018 IEEE Outstanding Large Section Award. This is the highest honor for the section, and this is a great honor, humbling experience to receive the best WA student branch, best student in Region 10, best young professional in Region 10 from 2019 President Toshio Jose Mura, future President Toshio Fukuda, and all Region 10 directors, past director. Let me tell you why I do research. I do not do research just to publish in high impactful journals and conferences. Actually, I do research to improve the community, to connect it to the sustainable development, to really touch the life of billions of people. In 2020, our EMBS region chapter has received Regional Outstanding Chapter Award. We have connected the world-class speakers to our young generation so that they can get help for their PhD studies and their research directions, and for the young faculties can have a postdoc opportunity. This award, Regional Outstanding Chapter Award, only 2 got in the whole world. One is Bangladesh and another is Latin America. By this way, we have increased the visibility of these subcontinent researchers to the rest of the world, which is very important. And also, you know, we have researchers like Dr. Jayanti and a lot of researchers, but we have to make them visible. We have to recognize them. And another is, you know, signal processing chapter, for which I'm the founder. And in 2021, we have received the chapter certification, which is a very great honor as such a young chapter, because we launched our own conference, PIXCon, and we have invited many distinguished lecturers. We have arranged the winter school for signal processing. So I really request you to visit my website, www.celiashanaj.com. I think we can change the world if we can work together. And also, I have my work in progress where we are applying deep learning for automatic detection of ALS patient, you know, ALS patient using the EMG signal. And also, we had a huge opportunity to apply deep learning for from ECG or EEG signal for driver drowsiness detection. Not only that, we can apply deep learning for other healthcare applications like jaundice detection, bilirubin estimation based on the SCLARA image processing. So these are the important area where I'm currently working and publishing to really connect my people around me, because in the subcontinent we have similar problems, so our solutions should be unique. Let us work together so that everyone do not start from the scratch. Let us share the resources, and for that case, you know, this colloquium is so timely and so effective, and topics are so interesting, so that this colloquium can open up a new opportunity for doing a research. So let us help each other, and let us share our resources. Let us do a collaboration. This COVID has taught us that we just need a Microsoft Teams, a Zoom link, or Google Meet to break the barrier and to really go beyond the border for the progress of research and innovation. So with that said, I want to thank you, Dr. Jayanti and her team, for inviting me, for providing an opportunity to share the contribution of my students, my young student, I should say. And that's an example how undergrad students, even if you are an undergrad student, you can do wonder and you can publish impactful journals, and that will of course help in your future PhD study, and wish you all the best. Thank you. And if you have any questions, I'll be happy to take those questions. Take care and stay blessed.
Participants can unmute your mic and raise your questions.
Yes. Any question? Dr. Jayanti and others.
Yeah. Thank you for your. I would also like to share that the EMKS student chapter of KS College of Technology received the Outstanding Student Award from EMKS in 2017.
It's distorted.
Okay, I think my mic is in trouble. Is it okay now? Am I audible?
Yes, ma'am.
Okay, thank you.
So any questions from the audience?
Now it is okay. Previously
Okay,
it
thank you,
was
Professor
disturbed.
Celia, for your presentation and your time to this colloquium. Thank you all, and thank you for the participants. We will now break for the lunch session, and we will be back again at 2 p.m. sharp. And one more presenter is here today for us to share the session at 2 p.m. And she'll be the last speaker for today's session. I hope all of you will join back with us at 2 p.m. sharp. Thank you. Thank you, Professor Celia, for your time. Thank you very much.
Thank you very much for your invitation. Take care and stay blessed.
Thank you. Same to you.
Jayanti ma'am, shall we begin the session?
Yes, ma'am. We can start.
Yes, thank you. Good afternoon and warm welcome to the speaker of this session, Shazia Nasreen, PhD scholar, IIT Kharagpur, and the participants of this international colloquium on healthcare and medical engineering. I'm Banu Rekha, Associate Professor, Department of Biomedical Engineering, PSG College of Technology, Coimbatore, and Secretary of IEEE MBS Madras section. I take this opportunity to express my sincere gratitude to Dr. Jayanti ma'am, who has invited scholarly scientists and academicians from all over the world for this international colloquium. So we had 4 such eminent speakers. We heard 2 eminent speakers in the morning sessions, and now we have Ms. Shazia Nasreen, research scholar from IIT Kharagpur, for giving us this session. I'm glad to welcome you, Ms. Shazia Nasreen, and I'm also glad to give a brief introduction about the speaker. So Ms. Shazia Nasreen is a researcher from IIT Kharagpur who is working in the field of visual cognition. Her work involves understanding how young adults and dyslexic children process different kinds of visual information. She is a clinical psychologist and registered with Rehabilitation Council of India. She has publications in Indian Journal of Clinical Psychology and IEEE publications. She did her fellowship in psychology from Tata Medical Center, and in her spare time she loves to read and travel. So with this brief introduction, I welcome Ms. Shazia Nasreen to this colloquium, and please take over the session.
Thank you, ma'am. Thank you for the introduction. Thank you for inviting me as well. Good afternoon, everyone. I'll be sharing the content and we'll start it. Is it visible?
Madam, not it.
Is the content visible? Can you see the presentation?
I'm not able to see. Yeah, it's appearing now.
Okay, today I'll be speaking about my research area. So I had presented a paper in EMBC conference on decision-making processes, how eye tracking can help in decision-making processes. So before I go into my research paper or our teamwork, I want to introduce you to the basic concepts of oculometric assessment and visual cognition. Again, I'm introducing myself. I'm Shazia. I'm working under the supervision of Dr. Rajlakshmi Guha in Cognitive Language and Information Processing Lab, IIT Kharagpur. Before going into what visual cognition is about, and there's a hype about visual cognition everywhere, we need to know what is visual perceptual processing is all about. Visual perceptual processing is actually defined as a mental organization. Visual perception is a mental organization and interpretation of that sensory information, which will help to understand what objects we are perceiving or events happening around us. So it is just a single process, or it is different processes are involved in the single thing. So there are different theories, as I said, but in Warren 1993, in his American Journal of Occupational Therapy, he has given a triangle with different processes involved, hierarchy triangle, from oculomotor movement, it starts from oculomotor movement, with visual acuity in a proper visual field, which will help to, attain the thing. When you have a proper in a visual field, you attain the thing, and when you attain the thing, you can scan it. And once you scan the image, for example, in front of me, there is a jug. The jug, you are attending to the jug. For that, you need to scan the jug. Okay, there is a handle and its shape is like this. And then you can understand, okay, it is a pattern. There's a pattern formed with it. And this pattern, when you match it with your schema or template, memory template you have in visual memory, it will help to understand, okay, this is known as a jug. So this is a jug. Now, if I have, this is an example I'm giving. If it is a problem situation, there are a lot of things happening. I have to take a decision. Then further after matching it with the template, I have to infer something. For example, I have to solve something. Then visual cognition comes in the role. So visual cognition is the highest at the hierarchical level, which will help one to understand what decision to be made, how to be made. And this thing, if visual cognition will help us to understand how different things we interpret in our life. So how do we measure visual perceptual processing? Is there anything specific? There are different biomarkers to it to understand the processing. There's no direct measurement of visual perceptual processing, but there are different parameters or different machines involved in understanding the process, starting with eye parameter, oculomotor movement, as I told you. Understand oculometrics. It can be through static eye tracker, mobile eye tracker. There's so much advancements in eye tracker nowadays. So you have different variety involved. So for example, you use static eye tracker when you have a screen and you have an image and you want the person to look at the image and understand, and you want to see how the eye movements occur in that image. But when you are for example, in a playground, you want to see how childs interact with other children. Where do they look at? So then you use a mobile eye tracker. So depending on the situation, you use the type of eye tracker. Now coming to brain imaging, there are different types of brain imaging which helps us to understand the perceptual processing through the cortical arousal level in the EEG or fMRI. Advanced levels are there. And I'm being from the psychological background. We know that in neuropsychological tests we have where we can tap different neurophysiological measurements, including visual cognition. So Bender-Jenstall is one of the examples. Coming to the eye movement in visual perceptual processing. Why I'm saying that, okay, I'm focusing on eye movement patterns here in my paper, because it is one of the important, if you can see the literature here, it is one of the important ways to assess visual perception. It helps to scan and analyze information. And it is as Varick said, that it is a principal pathway to understand perceptual processes. It helps us to understand where I'm locating my eye at, what I'm looking at, what the sequence I'm following. Then there is a Carpenter's idiomotor theory, which explains that eye movement may act as an indicator of one's immediate response to his environment. So at least what is happening at that moment of time, what I'm looking at, how I'm interpreting the thing, through the oculometrics, I can understand some of the processes. I was talking about defined images, as I said. No, I spoke about visual perceptual processing and how eye movement helps in that. Now coming to when I'm looking at defined image. Now visual stimulus can be varied type. It can be a defined with a proper contour, or it can be undefined with an abstract background. There's no such background. Figure and ground are not properly divided. So when I'm looking at defined image, what are the literature we see in eye tracking? We see that complex images, target objects involves longer to scan. We look at the edges at times, and when we are looking at a symmetrical image, we look at it unilaterally, and when there is a bilateral image, we look at asymmetrically. So eye parameters help in understanding associated cognitive marking as well as visual reasoning while perceiving an object. So we can understand, okay, for a defined image, there is a contour. We can understand through the contour what the object is, so we scan accordingly. Now coming to something abstract or undefined, there's unpredictable contours. So the fixations occur again at the unpredictable contours to resemble that to our schema memory, what we have, the template we have in the mind. Is it resembling to something like that? So we, for example, if you're given an abstract image, we try to obviously put that into something we know it. So we always try to relate it with our experiences. So we try to focus on whatever information we have. So even if it is unpredictable contour, we try to look at that positions and try to interpret. Rorschach is a very good example if somebody from psychological background is there. Rorschach ink block test is a standardized abstract image. There are studies where we found that fixations are mostly occurring at the central region. More fixation duration is occurring when the image are colored than, when monochromatic it is. And as it is written here, number of fixation increases with the presentation of card. The more the cards you show, the number of fixations increases with it. So I'm also working on this same thing. Our lab is working on Rorschach and as an abstract image. And later I'll explain about it a little bit after, completing what I'm going to say, what I presented in EMBC. Coming to eye tracking, I'm giving you little gist about what is all the processes about, so that you have, you can comprehend the paper much more easily than. Okay, fine. Coming to eye tracking, it is a technique to record where an individual is looking at any given point of time and what sequences it is follow. So what happens, if you can see in this image, this is our lab setup, and this is the computer we use. This is the Tobii eye tracker. It is mounted here. So from here, the person looks at, when the person is seated in a position, then we take the corneal, this works on the principle of pupil-centered corneal reflection. So what happens, the vector found between the cornea and the pupil, that measurement it takes to form a gaze. And then it is the external drive where the measures are there, and the sampling rate is 120 Hertz. Accuracy is 0.5 degree. And the best part is in the static eye tracker, there's no physical contact with the subject. In the mobile eye tracker, in the one you have to wear that. But in static, you don't have to wear anything. It is very comfortable. It is not intrusive at all. Coming to eye parameters, as you can see, these small dots are there. When you look at an object, it is written here, when you're looking at any point of time, at any sample you're getting, it is a gaze. And the gaze, when it is formed for a certain period of time, few seconds, it is fixation occurs. The whole thing comes as a fixation. And when you jump from one fixation point to another fixation point, saccades occur. So if you can see, fixation and saccades are happening accordingly. There are other parameters also, microsaccades are there, scan paths are there. I'm not going into it because our paper will deal with this only, gaze, fixation, saccade. Coming to my research, visual cognition, as I said, my paper was on oculometrics, application of oculometrics on visual cognition. So visual cognition is the process of making inference. As I told you in the first slide, it is primarily decision-based analysis. It helps to understand how we are preferring something over other. Why it is especially used in market research a lot, but it is not seen in day-to-day in learning situation how this helps. And eye tracking parameters such as scan pathway and fixation helps to understand the decision-making processes. What is the motivation of this study? Since there are studies which have already explored this feature of what decision-making, which eye parameters are related to it. What we wanted to see that how machine-made, human-made, and as I have highlighted here, images are human-made and create bias when the machine occurring. When we are looking at the machine and giving, it's not a machine-made, biases occurs. So we have used Clever datasets, which is AI created only, and it involves reasoning, and it has it conflicts multiple sources of errors. So we wanted to show how an AI-based questions help one to solve problems, or what are the processes involved in it, how the person is looking at an image. Already the AI has solved the problem in the best possible manner. Basically, they use the Bayesian model. So now I want to say, when human has suboptimalities in their decision making, apart from just looking at an image or the fixation occurring, there are many multiple suboptimalities occurs, which is a psychological process. Can we relate this to the machine-made questions, and what are the factors involved in it? The work, as I say, what is the work? It is to study the effect of complexity of task on visual cognition. What is the complexity of task? complexity of task can be explained as two things, as I'll be explaining further. It is the number of objects present in an image, the semantic nature of the questions involved. Semantic nature of the questions involved, meaning some of the questions are twisted, they're not directly asked. So when a participant is given such questions, how the person is solving the problem, which areas they are looking at when they're trying to, answer those two type of questions. It can be varied from simple to complex one. The performance parameters which I have taken is number of correct responses and total response time. I parameters which we have taken is time to first fixation duration, number of visit counts. Ethical approval has been taken from the institute. So the sampling technique was purposive sampling technique. So we have taken students from IIT Kharagpur, 30 engineering students, and the inclusion criteria was that they should, since the questions were in English, already published in English, so that the students do not face any problem. So we have taken only from the students who have the background from English medium school, and there are no prior exposure to these questions before giving it to them. And of course, they are the students of IIT Kharagpur only. Exclusion criteria, if there is any eye related problem like PAR also, sometimes the proper points are not calibrated. So we have excluded such students, and students with any psychiatric disability or unstable mind, we try to exclude those sort of students. So 19 males were taken, and for 11 females, they were of the age of 21 to 30 years. So coming to the Clever data set, these are the questions retrieved. This is known as compositional generalization test. They have presented this paper in a conference, and where, as I said, it is a AI based, questions. AI have developed the questions on their own, which again includes, reasoning. They have tried to include that human reasoning process. They have tried to include in the AI machines also. And now I want to see in my paper, or our team wants to see, is how humans solve this AI developed questions. So there are many, as you can see, there are a lot of images, 70,000 images with 6,99,000 questions and more. So we just selected 4 images with varied complexity level, starting from the simple 4 only objects are there, as you can see, and it increases with the number of the objects, and they're all clustered together, some are paired. So different complexity level were kept into mind, and this were randomly selected. The images were randomly selected. So for example, if I give you if an image is presented like this, and there's a question below that, if you can read, is there a small yellow object of the same shape as the big yellow object? So the first thing is that I can see there are 10 objects clustered together of different shapes and sizes. Being how my processing will involve. The question is there a small yellow object? I have to look at the first thing, how I'm solving it. I'm looking at the size. Okay, is there a small? Small is the size, color is yellow, shape, I'm saying same object of the same shape. So I have to look at the shape again, and then I have to compare with the big yellow object and see whether it is present or not. So I am going through different levels to answer a particular question. So this will help me to take the decision. So where my eye is looking at, I'm fixating my point at to answer a simple question. Some people try, there is AI, since it is AI developed, AI has its own way of, giving correct answers. So it has its own model and interpretation. We wanted to see how human can give correct answer with looking at this. In what pattern it gives, which area they cover to give the correct answer. So as you have seen, there are 4 images. From that, we have taken one easiest and one most difficult question for this EMBC conference to see how these images affect. Easiest one is, if you can see, there are 4 images, and this is the easiest question out of the other question. Is there any other thing of the same color as the big cylinder? There's a big cylinder. Is there any other thing? I have to just look at the presence and the shape. As well as size, yes. And most difficult is that what is the shape of the cyan object left of the big metal cube that is in front of the tiny gray metallic thing that is the right of the small cyan block. So I have to go through different processes, and this has been twisted at the semantic nature has been more complex. So it is more complex in nature, and it came out to be the most difficult question. So the experiment was like this. The questions were shown in that computer screen, and the protocol was that 4 images were shown. Each image had 6 questions, and then sequence bias was given. They were not given, all were given, not given same questions. They were biased. To remove that biasness, we have done IP1, IP2, IP3, IP4. There are four patterns we gave. In 4 different patterns we gave the card so that we do not create any bias of giving the same in same position. Procedure, coming to the procedure, participants, firstly, the aim, what eye tracker is all about, what they are going to do. Those participants were briefed about the experiment. Second, they were informed consent when taken, whether they wanted to participate or not. And after taking their consent, step 3 involves following a mark on the screen. That is static eye tracker were calibrated with respect to the participant side. So you can understand they had to sit in front of the computer monitor, and their eyes were calibrated before the experiment started. Next is the last step, step 3B, where the display of visual reasoning questions like the clever test questions were shown to them one by one, and they need to answer it out. No time limit was set. We wanted to see how they give the correct answer, in what time span. Participants' eye tracking response to the stimuli recorded by the eye tracker. Participants' verbal response were also recorded by hand. What answers they were given, correct or not, we were also writing it, like one of our members were writing the answers. Analysis was that each participant was assigned a score. Time to answer also recorded through the software. Gaze recorded by the software, fixation and other metrics were calculated, and then we did the correlation statistical analysis. This flowchart is all about how we have analyzed the scores. We found that high complexity question is related with high conceptual difficulty, and then obviously longer fixation duration was related with it. Back tracing, when the question was complex, the person was looking at it again, going back, moving their eyes from the question again, coming back to the question to confirm whether I read the question correctly or not. So it is depending on the semantic nature of the question. The differences in eye parameters are validated by individual task performance also. Significant differences were found between like when the number of correct responses given, the more the complex is, the more time they have taken to answer it, and high chances of errors were there. So if you can see the image, I have given just an example for question number 2 and 424, the most easiest and the most difficult questions. If you can see how the heat map and the fixations occurred here in the images. Go below, if you can see the question number 24, there's a lot of fixations occurring and the heat map occurring. So the more the complex the questions, the more fixations is occurring. So what we got from this paper? This data is being applied to develop a computational model. Right now we are working on, our team is working on developing a computational model for reasoning and decision making. By using a spatio-temporal framework. Human decision-making errors and memory limitations will be incorporated because there are so much suboptimalities in human perceptual decision-making. As Rahnev and Danshan has mentioned in 2018, there are different types of suboptimalities which occurs in human decision-making. We wanted to create something similar to it, how a person is responding when they are looking at an image, keeping in mind the suboptimalities. So we are developing a model for that. So this is my EMBC presentation, and my team members, I would like to introduce them. Dr. Rajlakshmi Guha, Professor Rajlakshmi Guha, Professor Jiyal Paik, my senior Anup Kumar Rai, I, Prachi and Nikki, we all worked on this, and some of us are still working on this. This was the paper which I showed you. I presented in the EMBC conference. Since I have time, I think. Do I have time?
Yes, yes, Michelle. Yes.
So the ongoing how we perceived undefined images. As I told in the while introducing the slides, I told you there are different type of images. We till now spoke about how in a defined image, how high complex situation person is responding. But what happens when there is no defined contour is there? So how we are responding in that situation. So this I'm just giving a brief about the walk, very brief. We wanted to see that how an abstract image can be perceived, which areas draw more attention to respond. Since there is no defined image, there is no contour, there is no figure in the ground, which areas with stimulus, with their ages, there are studies, but there are not a defined studies, which areas actually activate our attentional processes. For example, what happens in Rorschach at times, person is looking, for example, fixating their eyes at a particular position, but they are saying it is a bat for the first card. So is a single area responsible for saying the whole bat, or there is a scanning occurring through the whole image? So we wanted to know whether bottom up or top down processing occurring, how the images are scanned, how we are understanding that a simple abstract not defined image is given meaning. So which will also help us to create different study materials later on. I'll come to that. So study approach is that, as I told you, holistic versus sequential approach, saccadic movements occurring within an object, fixations and scan paths happening. Methodology same, I'll be using eye tracker. I'm using eye tracker. Rorschach in blood test, as you can see the cards, there are 10 cards are there. They're abstract, they have no defined meaning with them. There's nothing right and wrong. And the procedure is that we obviously have an ethical clearance for that procedure. 25 young adults were taken, and those who already had previous exposure to Rorschachs were not taken because they would give a biased response. They will know obviously, okay, this is resembling to that. So we want only people with no exposure to Rorschach. And after that, we wanted to see that applicable algorithms related to it, and how an image is perceived, which areas are contributing, how the scanning is occurring, what type of scanning is occurring. So basic ones which we have done, paper is published in EMBC only, is fixation density, initial fixation, and the SF ratio. SF ratio, which parameters they are. So for example, I'm looking at okay, I'm showing you. Yeah, here it is if you can see, these are the images. So in the heat map, if you can see, before going on to what I was going to explain, the marked divisions, I will talk about this, the heat map. As you can see, mostly the card, if you can see, the central focus is there. When we are looking at a defined image, you saw that the boundaries were looked at and then came to a central position. But here, if you can see, mostly central fixation is occurring. And these are the two cards. This is card 2 and this is card 1. And also, if you can see here, this one in the different marked regions, which areas are activated, we want to see. For example, here, 2D4, D5. I can actually increase this, zoom it. There are different images with that. So there are different marked regions. They are according to the Clover protocol, and some are used, algorithms were used and developed. So there are two different type of scanning ways we have developed. Our team has developed, and we found that some areas is stimulated more to answer a specific answer, to give a specific answer. For example, two-winged enamel. The person doesn't have to go through the whole scanning. They just have to see the essential areas like D2, D4, D5. The essential area person looks at to whoever has given this response has given dispositions. So we can understand which areas are activated when they are looking at an image. I want to conclude the whole thing. Visual cognition is a functioning which is actually what is happening, who is happening, you're getting an information related with your past experience, which color your outcome. But how we will understand it, we need something which will help us to understand to it. As I say, there are many types. An objective marker, which is non-intrusive, is ocular movement through eye trackers. With the advancement of eye tracking technology, one can validate one's research through the visual psychological processes can be validated through this eye tracking technology, and we can create a better living. Why I'm saying a better living? Because once we know how these images are perceived, how different informations are interpreted or scanned, we can actually help students with learning difficulties like dyslexia, how they interpret information, how they, while we can't perceive the normal concrete alphabets, they are creative. So whether abstract informations are more useful to them. So through getting this type of information, we can create a model which will help us to also develop a learning material for this kind of children. Also, we're understanding the cognitive load. This is very useful in human-computer interaction, how we can use this information to, either increase or decrease the cognitive load of the students, or give it, proper understanding according to their age and factors what are all involved. So being in this coronavirus situation, we have to be at home. So we need to have, specific developed proper learning material for this kind of children, especially where students cannot go to schools or colleges right now. So the references, and thank you. Any suggestions?
Thank you, Ms. Nasreen. It was a very informative talk. We got to know a lot of inputs, and you took us through right from what is visual cognition, and then you showed us, the methods that are used to arrive at certain parameters. So I have one basic query, Ms. Nasreen. Like you said about the fixation and the another term, I'm not sure, started with S. Saccades. So how are they quantified? So does your iGrace software itself gives you about the fixation and saccades?
Yes, they have like the Toby we are using, they have their own software for that. Also, our team members are there who are from the computer science background. They also develop different algorithms. For the Rorschach, I have my colleague who has developed that type of different scanning. One, we are getting the information from the software itself. In Toby, you will get an information. But if you want to see something specific, how the scanning is occurring, you have to develop an algorithm for that specific thing which you want to. And then you can, apply to that model.
Okay, that's great. And you have said that this study can be, using the outcomes of the study, we can assess what is happening to the attention span of dyslexic kids, if I'm right. So
we can, ma'am.
Yes.
Yes.
So is there how do you plan to expand this? Like, will you recruit dyslexic kids for this, or will you reach out to some institutions?
Yes, ma'am. Yes, ma'am. My plan is to work with dyslexic children with this information, because what happens, these are the children whose visual perceptual field is affected rather than from other children. They look at an information, but they cannot take in. So there's a basic neuropsychological theory of magnocellular theory and how the brain works with it, and that is affected in them. So what type of information, when we present to them, in what aspect will it help them? So we thought to use this abstract image, defined image, some script, some we want to use also symbols, and see how they perceive these images, like the processing occurs, oculometrics involved in them. So we can use this information where we find them, okay, they are performing better in this, so we can relate them, and later on we can develop a model for learning material according to that model.
Okay, that's great. That would be really needed for the society and the kids who are
our team will work.
Yeah, thank you, Ms. Nasreen. Any questions from the participants?
Participants can unmute your mic and raise your questions.
Participants, any more questions?
Thank you, Ms. Shazia, for your acceptance and presentation. This is Jayanti. And that was a nice presentation. Am I audible?
Yes, madam, you are audible.
Okay.
Sorry, I was not. I had unmute. Sorry. Yes, ma'am. Thank you so much. I had interaction with you through mails. Yeah. Thank you.
And wish you all the best for your thesis submission.
Thank you, ma'am. Thank you so much.
Best wishes, Ms. Nazia.
Thank you, ma'am. Thank you so much.
Thank you. Ma'am, over to Jayanti ma'am for the remarks.
Yeah. Okay. So if there are no more questions, I think we can close today's session. And I request all of you to be to join us tomorrow at 4 p.m. because you can see the schedule which is displayed. The first talk is by Professor Ganesh Nayak of Flinders University, and the second is Professor Michael Fu, Electrical Stimulation and Virtual Environments for Neuro Rehabilitation. It's all hardware oriented. The undergraduate students will definitely have an interest over this. And then emerging applications of medical ultrasound, which is about image processing. And the last talk is medical image analysis. So join with us tomorrow 4 p.m. and it will go till 6:30. Join us, stay tuned and listen and get benefited. Thank you all for joining us today. And my heartfelt thanks to all the speakers who have presented their papers. On behalf of Madras section, IEEE EMBS Madras chapter and KS Rangasamy College of Technology. Ma'am, you have got anything to tell, ma'am?
Ma'am, thank you. Thanks to all the participants. Thanks to the organizing team headed by Jayanti ma'am, who has done an excellent work, ma'am. My sincere appreciation. Thank you.
Thank you, ma'am. Thank you very much. We will meet tomorrow. Please do connect at 4 p.m. sharp. Thank you.
Thank you, ma'am. The session is officially ending.